चलो उतरो 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 बैठ के साई बात यार ये रिश्ते बना रखे हैं पाप रखा है इससे I request the technical team to please allow Dr. Sairam. He is in the waiting. देखो बढ़िया है ये समझ
Hi, this is Saidam. Can somebody confirm that you can hear me?
Yes, sir. We can hear you. You're audible, sir.
Oh, great. Thanks for that goof up.
You're welcome, sir.
There was some issue. Okay, so we'll start.
Yes, sir.
Kritika madam, please.
Yes, sir.
Please start the program.
Yes, sir. So good.
Start.
Yes, sir. So good afternoon, ladies and gentlemen, esteemed guests and conference participants. Welcome to DBT-CTEP, Government of India sponsored National Conference on AI-driven design and development of new chemical, biological entities and formulation approaches, challenges and road ahead, organized by School of Pharmacy, DRIEMS University. I am Kritika Mahanti, Assistant Professor from Department of Pharmaceutics, School of Pharmacy. You are host for this scientific session. And it is a truly pleasure to hear with you all. So today's session is about learning, sharing ideas, and understanding how artificial intelligence is shaping the future of drug discovery and development. I hope this time became meaningful and inspiring for all of us. So without any further delay, let's move ahead with our exciting schedule. Now it's time to privilege. Now it's time and my privilege to introduce the session chair for this scientific session, a highly respected academician, researcher, and visionary leader, Professor Dr. A. Ramakrishna Savarya, sir. Dr. Savarya is an inspiring personality with remarkable achievement in the field of pharmacy education and research. He completed his B.Pharm and M.Pharm with excellent grades from Mangalore University, followed by an MBA and MD in alternative medicine, and later earned his PhD from the Tamil Nadu Dr. MGR Medical University. He has served in several prestigious academic positions and has been leading Srinivas College of Pharmacy as a principal and director since 2008, with more than 320 research publications and over 1,500 citations. He has also presented more than 80 scientific papers at national and international conferences and had guided a large number of M.Pharm, PharmD, and PhD students. In addition, he has contributed significantly through his involvement in various academic bodies and editorial boards and university committees. Dr. Savarya has received several prestigious recognition, including Indian Drugs Best Paper Award, Young Presenter Award, National Educational Leadership Award, Principal of the Year Award, last but not the least, the Eminent Pharmacist Award 2025 from the Government of Karnataka. Sir's dedication, achievement, and contributions truly make him an inspiration to all the pharmacy professions. So, ladies and gentlemen, please join me in this welcoming two eminent personalities. First is our chairperson for the scientific session, Professor Dr. A. Ramakrishna Savarya sir, and our distinguished speaker, Dr. Sairam Kalpatapu sir, an expert in medicinal chemistry and computational chemistry and bioinformatics. He is be delivering a highly inspirational talk on antibody discovery in the AI era. So now I request our session chair, Professor Dr. A. Ramakrishna Savarya sir, to kindly introduce our distinguished speaker to our delegates. Over to you, sir.
Thank you, ma'am, for your introduction.
Thank you, sir.
To begin with, I acknowledge the thanks to the organizing committee, Siddharth and Amulya Ratna and team of this School of Pharmacy, Dream University, for giving this opportunity, organizing this
DBT and CTEP Government of India sponsored this conference. And I take the pleasure of introducing Dr. Sai Ram Kalpataru, who is going to deliver a talk on this antibody discovery in the AI era. Dr. Sai Ram Kalpataru has done his M.Pharm in medicinal chemistry from Rajiv Gandhi University of Health Sciences, Bangalore, and PhD in computational chemistry from Jamia University, New Delhi. He worked over 2 years in Deccan School of Pharmacy as lecturer and moved to GVK Biosciences, Hyderabad. In 2006, he moved to Ahmedabad to join as head of the department bioinformatics, Jaides Research Center. There, he contributed to identification of novel targets for dyslipidemia and metabolic syndrome. He is currently working at Sun Pharma Advanced Research Company Limited, Vadodara, Gujarat, as head Department of Bioinformatics and Computational Discovery. Here, as well, identified novel targets for cancer and spearheaded integrated drug discovery teams towards identification of potent inhibitors. He published many research articles and patents on his credit. With this brief introduction, I welcome Dr. Sairam Kalpataru to give the more information on this interesting topic, that is antibody discovery in the AI era. Welcome you, sir.
Thank you, Professor Ramakrishna. At the outset, thanks to Professor Amulya Ratna Behra and Professor Siddharth and Kritika Mohanty for giving me this opportunity. I think, I'll share my screen. Are you able to see my screen?
Yes, sir.
Yes, sir. Yes, sir.
Okay, great. Okay, so I'll start my discussion on antibody discovery in AI era. Specifically, I'm not a biologist to start with, but having worked in this area and having seen the practical utility of some of these tools, I feel myself, fortunate to share my experiences in working with some of these tools. So what I'm going to discuss today is setting the stage. So I'm not very sure, how many of the delegates who are actually attending are into core of antibody discovery. But I would rather set this stage so that, right from undergraduates, graduates, postgraduates, and PhD students, everybody can understand what is the current state of discovery in antibody area. And some of the models that are currently are used for discovery of novel antibodies, and what are the challenges, and finally summarizing our learning for the next 35 minutes or so. And we'll reserve last few minutes for question answers. Wherever I can certainly give the answers, I will contribute. So to start with, as I said, setting the stage. So now the structure of the antibody, there are multiple of them. There are many types of antibodies like IgA, D, E, G, and M. But since the IgG is most common variant with high bioavailability and cell permeability, so this is something which most of the antibodies have their core structure as. So the polypeptide antibody has two identical heavy chains and light chains held together by disulfide bonds. These are disulfide bonds. And the two arms of Y are called FAB. This is FAB region, and this is fragment crystallizable region, that is FC region. So the cytotoxic activity of given any antibody is coming from this region, and then antigen binding region is coming from this region, which is also called as, which is part of FAB region, also called as FA. So there are two regions within this, that is heavy chain and light chain. So as we go ahead, we'll also see other intricacies that are involved in understanding some of the anatomy of this antibodies. So some of the popular antibodies that are approved are given in this list. As you can see, from almost like 15 odd years, you see that almost about 30 monoclonal antibodies are approved. So which essentially means that they are really picking up the pace, and almost per year there are a couple of antibodies that have been approved up till now. So going forward with the more latest technologies, I believe that this would be picking up much more pace than current speed. And I just for the sake of audience, I've given how these antibodies are named. So some of these things are given, so I think you can read them by yourself, but we'll progress why AI technologies have to come into picture, what are the limitations of some of the current technologies, and where the physics based models actually contribute. And I'm going to keep this entire session a top level thing, because half an hour 45 minutes are totally insufficient for any single topic to be covered in complex topics like monoclonal antibodies. And on top of that, I'll not be able to do justice for a single topic if within this time frame. So that's why I've kept it a top line summary of what is actually happening, where the current challenges are, and the limitations of the current model, and where the progress is happening going forward. And I'm also not going to cover anything related to AI per se, like if it is an autoencoder or say transformer technology or deep learning technology. I'm not going to go deep into details of any of these methodologies. Again, each one is a subject in itself. So with that disclaimer, we'll move forward. So usually experimentally directed evolution is, or rather antibody discovery through directed evolution is done by two different techniques. One is phage or yeast display technique, and another one is immunization of animals. Now there are limitations to especially these two techniques, is that difficult to target specific epitopes on the antigen. So antigen is the target. Say for example, if we say EGFR is our target, okay, so the antibody which is actually binding onto EGFR can bind in multiple ways. So EGFR ecto domain can have multiple epitopes. So if a antibody has to bind to one epitope, it has to have interaction with certain regions. So, or if it has to bind to some other epitope, then there itself it has to bind to multiple regions. So that way it is difficult to target specific epitopes on the antigen, because a single antibody, when it is coming out of yeast display techniques or immunization of animals, they can be non-specific as well. So that is the reason why it is difficult. And only a small subset of antibodies has biophysical and other developmental properties required to become therapeutics. So what are these biophysical methods? See, biophysical methods are actually measuring whether the given antibody is specifically binding to the target intended and to the epitope that is intended. So that is what is biophysical. So 1:1 binding is what is being seen in biophysical methods. And the properties that are required to become any antibody as a therapeutic is that some of the bare minimum properties like solubility in high concentration and scalability and non-aggregating potential, non-immunogenic potential, so on and so forth. So some of these things actually become very important for any antibody to eventually become a drug. And antibody structure information is so important for understanding the antigen-antibody interactions, because if this is not assessed, then each antibody may have non-specific interactions with other epitopes, which can lead to side effects, just like how we understand in the small molecule drugs. And out of 6 CDRs will come in the next slide. What are the CDRs and how they are, what are the difficulties with some of these CDRs and what are the advantages with some of these CDRs in the next coming few slides. Best hear me out for this. Out of 6 CDR, there are 6 CDRs. That is, on the heavy chain, there are 3 CDRs. On the light chain, we have 3 CDRs. So, but out of 6 CDRs for H1, H2, L1, L2, and L3, we have limited structure conformation variability. So these are actually basically these CDRs are loops. So there is no particular structure to any of these CDRs. Since they are loops, there is a possibility that a loop can be like this, a loop can move like this, like this, like this. But within H1, H2, L1, L2, and L3, there are possibility of limited conformations. Okay, so what is difficult is H3. So that's what we are coming to. CDR, that is conserved domain region, H3 loops have high conformational diversity. And more complexity comes when in cases where high similarity sequences also has significantly less constraint. So it means that if CDRs of one antibody and the CDR of other antibodies, if the sequence of CDR3 is very similar, in spite of that high similarity, it can have significantly very high conformational variability. So, so from this particular slide, we are learning that traditional antibody techniques or discovery techniques like phage and yeast display techniques or immunization of animals have limitations, and those limitations we have discussed. And then need for biophysical one-to-one interactions are essential to understand the specificity and affinity. And developability properties are also a requirement. And structure is very, very essential to understand, to further fine tune the specificity of the antibodies. And the recognizing motifs on any given antibody with the epitope or the antigen part are 6 regions on the antibody. They are called CDRs. The 5 of them have limited conformational variability, while one, that is CDR H3 loop, has very high conformational diversity. And the entire crux of the AI or physics-based modeling lies in understanding the conformational flexibility of this CDR H3. So this is the anatomy of exactly what we are talking about CDRs. But before that, we will revisit the FC region and FAB region, and this is FV region where the CDR regions are present. These CDR regions, this is the light chain, and this is the heavy chain. Okay, so the heavy chain CDRs are H1, H2, H3, or CD1, CDR1, CDR2, CDR3, and the light chain ones are L1, L2, L3, or CDR L1, CDR L2, and CDR L3. So these are the regions which are actually interacting like this. Okay, so this is the antibody. So this is Y-shaped structure, and this red region is the paratope. That is the region which is actually recognizing the epitope on the antigen. So this is the target protein where this interaction is actually happening. So now let us zoom in on to this region and try to understand how these are oriented. As you can see, this is the variable light chain where you see that L1, L2, and L3, that is on this chain, the CDR region. And this is the heavy chain on which you see H1, H2, and H3. And you can notice, can you see my pointer? So you can see that there is H3 here. So which is a bigger loop, but still, there are a lot of conformational variability that is coming out of this region, and this is very important for the antigen antibody interactions. So if this is clear, we'll move forward and see what are the different techniques and their limitations. So many, as we have said, that many experimental antibody techniques yield sequence information only. So with the advent of next-gen sequencing techniques, so we see that quickly we can find out the sequence information. So once an antibody is generated either from immunization technique or this phage display techniques, so the protein or the antibody is isolated and that can be sequenced. So many experimental techniques yield sequence information not associated with the structure. So basically there is no structure involved at this stage, only sequence information. And independently determining all antibody loop structures is a laborious and totally impractical for any organization. So nobody does this. So when these DL methods or other AI methods were actually developing, so there is another approach which was computationally addressed based on physics. So they are called physics-based modeling or molecular dynamic simulations, or based on multiple sequence alignment and things like that by using homology-based models, or something called as modeler, where both approaches have been used. So these are some of the conventional physics-based approaches which are generally used. So moving forward, I will share my own experience with one of these approaches very quickly. But my only concern is that I'll not be able to disclose any of the target information, neither the intricacies involved there. But I'm going to discuss with you what exactly has happened, the methodology, which is the heart and soul of any approach. You can use the same approach for discovering the epitopes for given antibody for any target. Okay. So this is based on a target which we were working at Spark. So what did we do was, we knew the antibody. We in-licensed this antibody from a US-based academic institution. So they knew that this antibody was binding to a particular epitope. But what is not clear was that where it is binding and how it is binding. So the problem came to us that, can you actually find out where it is binding? So that if they want to do any modification on the antibody, they can actually go ahead and do it. So what we have done was, since we know that this antibody is binding to the target that they have disclosed, what we did was we have simply used a protein-protein docking, and then we did docking of this molecule, this antibody, onto the epitope. So now, as any antibody, as any docking tool does, this antibody was picked up and it was put onto different regions of the protein. And then multiple conformations of that were given out as an output. So now once we have identified that, we have evaluated intermolecular interactions like hydrogen bonds, aromatic interactions, van der Waals interactions, shape of the binding site where this antibody is binding onto the antigen. So those all were collected into a table, and then we said that what are the each and every interaction energies, their distances, everything was captured into a table. Now then, after that, once we know that where CDR3 is binding, so we have done amino acid mutations. I mean, imagine that this is your CDR3. We have made amino acid mutations on the CDR3 where this is binding to epitope. So once we made this each amino acid mutation and we redocked that onto the antigen, we saw there is a loss of stability. So which we have assessed by using molecular dynamic simulations. Now, like that, we have done for each and every amino acid that CDR3 was actually interacting with the antigen region. And then like that, for multiple of those poses we have evaluated, and based on the energetics, we said that most likely this antibody is binding to one particular epitope that we have identified after all this analysis. There's a very, very huge work. But unfortunately, because of IP reasons, I am unable to disclose, neither we could publish it. So when we have said that this is the region, these are the amino acids, so our biotech guys, they went back, they have they did the mutations on the epitope region, that is on the antigen portion. And when they have put their antibody, now it shows no binding. I mean, this is surface plasmon resonance, SPR study, which is a biophysical method where if the binding is there, there is a signal. If there is no binding, no signal. So simply, the moment the wild type was taken, there is a beautiful signal, and the moment the amino acids that we said are important for CDR3 binding for this particular binding mode, loss of binding. So this is one study, and this can be applied for anything. And there is, I mean, methodology wise, the way we have used is the only novelty, but rest of the all things are known from the literature. So we'll move to the AI and the other related approaches that we are going to discuss today. So DL models for antibody structured modeling. So there are a handful of models that are already known in the literature. This is antibody builder, then AB looper, deep AB, and IG4. These are some of the DL models that are already known and that have already been evaluated by many scientists across working in this field. So this antibody builder, it is an automated antibody homology modeling pipeline. It follows template selection because antibody has to be built, so you need a template. Orientation prediction, how CDR3s are oriented, how FC is oriented, and things like that. CDR loop modeling and the sidechain prediction. Where DL portion is coming is by doing ab initio modeling of CDR loop. So using some of those templates. And then AB looper, so it uses graph neural network, which can directly work on 3D coordinate data from the structure files to predict the positions of all backbone atoms. And deep AB, it uses deep learning to predict invariant features, which are then used for restructuring structures using Rosetta. Rosetta again is a University of Washington product for specifically for antibody modeling. There are models specific for antibody modeling. IgFold is a protein language like LLM. It is a protein language based model. It can predict structures faster, one structure under 1 minute time frame. And IgFold incorporates template structures, leading to more accurate predictions. If there are template structures that are available, it improves the accuracy, especially in the nanobody structures. So the current challenges of some of the computationally designed antibodies is that reliability of modeling of antigen antibody remains an unsolved problem. Now, up till now, what we are talking is antibody. So unless and until we understand how antigen is binding to antibody, so you really do not know what is the precise epitope and how it is binding, what modifications further on you can actually do on some of those CDRs and all that, we'll not be knowing. And the actual IP comes from the region which is actually interacting, so or close by areas. So that is very important to understand. So that reliability is something which is still has to be solved. CDR H3 loop, as we said, it is still a challenge, and improved architectures would subsequently improve model prediction accuracy. And CDR loops with similar sequence adopt totally different structural conformations. This point we have already seen it. And alpha 2, many of us might have seen the AlphaFold 2 publication, so which has come in I think in a couple of years back in Nature. So where they have clearly shown how the protein structures can be predicted using multiple sequence alignment and some of the transformers transformers technologies. So those things can be done using AlphaFold 2. And what is the limitation of this AlphaFold is that there is very little information about antibody structures or antigen antibody structures up till now in the experimental and the public domain. So that becomes a limitation. So of late, there are experimental methods which are picking up the pace, like cryo-EM technologies, which are actually which can actually identify the three-dimensional structure of any given macromolecules like antigen antibodies, and then we can understand their interactions. So yeah, so current work is to attempt generate structures from single sequence inputs alone to address the challenges of dependence on multiple sequence alignment. So as I said, the limitation of antigen antibody complex structures, if it is a limitation, then we need a technology which can give the antibody antigen structures or CDR-H3 loop modeling just based on this sequence criteria. So that is the current work which many people have started working on. And one of that is Equifold from Genentech. They do very well on the antibody structure. And this Equifold is again, it's a DL technology. And binding to antigen could lead to conformational changes. And it is critical to extend current models to include antigen information, and which is in progress now. Yeah, I think in a few more minutes. So DL-based DLAB localizes this interface of an antibody and antigen and use a 3D convolution-based neural network to generate binding scores. And limitation of such models is to include necessary discretization. So discretization is a plus and as well as minus. Discretization of how the antigen and antibodies interact, because this is going to be a very large surface, and to understand how antigen is interacting with the antibody. So DLAB localizes to the interface of antigen and antibody, but unfortunately this discretization sometimes leads to loss of information as well. Although this is trying to address such a large surfaces and the interaction regions and so on and so forth, but this discretization is also leading to significant loss of information, and that leads to potential inaccuracies in the models that are being predicted. So that is one of the limitation. And structure based models, they use variational autoencoders or things like that to overcome limitations of classical antibody designs, specifically CDR H3 loops. So those which are predicting backbones of H3 CDR loops by producing a 3D coordinates by using variational autoencoders, or models that predict structural energetic changes. That is, they are going to predict change in the change of free energy for redesigned CDR regions. If we find that CDRs are not proper, so if you want to change any of those things, then the change in the change of free energy, that is a physics-based modeling, where you will actually figure out how they are actually interacting with each other, and what is the change in the change of free energy with the change in each of those amino acids which are interacting. So sequence-based, as I said, since structural information is very limited, so sequence-based information is also important, and a lot of things are happening in that. So one of that is BERT architecture, which was directly trained on to UniRef 100. These are databases. BFD 100. These are data sets which have got millions of sequences, and based on that, a protein language called as BERT was made. And this understands textual information to figure out how the antibody may fold, and then things like that. And this is also ESM1 1B with 250 million protein sequences. It's also these protein sequences were used to train a language model which learned embeddings that encode important biological properties. So these are the two things that are actually picking up pace in this case. There might be more, but some of these things which I have gone through and I found interesting, I'm discussing. So, and both ProBERT and ESM1 were highly successful at capturing most properties of protein sequences. And there is antibody language model which showed strong performance in predicting missing amino acids in the OS database, where 40% of the sequences are missing at least 15 amino acids or more. So there are many sequences where 15 or more amino acids are missing in the sequence information. So for those kind of things, we understand kind of chat GPT. So similar to that, this is a gross simplification, but then for the sake of understanding, you can say that a blank model actually can figure out what are those missing amino acids, and actually you can actually impute those missing amino acids that it is giving out into the actual model. So there are reports that have shown promising performance in predicting binding affinity to 3D structures. So this is also an important thing. Usually, binding affinity is an outcome of experimental information. That is, you take a SPR, or you get these information from either an SPR, an ITC, or things like that. But then in this case, just sequence information also, people are actually trying to figure out how they can measure the affinity, or how they can actually predict the affinity. So the antibody model is an antibody specific BERT model, which was trained on 558 million antibody sequences with embeddings that cluster into directed evolution trajectories and the ability to detect Pareto binding residues. So essentially, this model is trying to figure out what amino acid sequence of the antibody will actually go and bind to what antibody sequence of the target of interest, and then what the target essentially is. So there are other language models like immunoglobulin language model, IgLM, and other natural language protocols or models that can be used to perform affinity maturation on even clinical grade molecules. What is also interesting is that without even knowing what an antigen or how this is actually looking like, so measuring the affinity is something which is like very, very surprising. And so some of those things are also in progress, like IgLM is what they have very clearly said. So this is a summary of all the computational things that we have touched upon. So I would not say we have not discussed anything in detail, and within the given time frame it is also difficult, but what I feel is that some of these things are useful for routine antibody discovery and optimizations. And number two is that physics-based modeling is certainly an option, or rather a very clear option to start with, because those are actually giving you the guidance to go forward. But in the case of any of these deep learning models, we still need to validate some of these things with experimental information, which I believe will be very scarce to or very scantily they will be published, because that's something which people will look forward to go and file their IP on. So I think physics-based modeling is still still an option for antibody designing, and some of these things can be evaluated and explored at local level. And to conclude, the key learnings that we have understood today are the limitations of traditional antibody engineering methods, and which inspired AI to design better antibodies. So that's the context. And antibody structures have conventionally been predicted using modeling simulations, or some of the hybrid methodologies like modeler. And DL models have been attempting to learn overall features of antibodies from their sequence alone, and in turn make models which can be predicting the CDR H3 loop and their conformations. And it is likely that future models would actually incorporate both features from language models, and subsequently train on experimental data, so that the optimized antigen antibody binding can be close to actual reality. At the moment, purely sequence based models are either antigen agnostic, that is, we don't know anything about antigen, or need outside experimental data, and would benefit from incorporating structural informations, so during generation process, which is again very expensive and time consuming methodologies. So I think that's what I wanted to convey, a bird's eye view of where this AI era is taking us in the antibody field. So I will end my session there, and I thank my company I thank my colleagues who have helped me in evolving some of these methodologies. And I'm open to take any questions if I can answer. Yeah.
Thank you, sir, for your enriching presentation on antibody discovery in the AI era. So now I request our session chair for clarifying any queries. Is there any queries? Anyone want to ask some questions? Kindly put it your question in chat box. Or anyone wants to ask question, you can directly ask question on the session. Unmute yourself.
Yeah, I think the topic is too easy to understand, or it is a bouncer. So either of these two. Yeah, Namit Naik. Yeah, you raised. Yes, please go ahead, Namit. No, I'm not able to hear. I think you have to unmute. Or probably, Namit, you can actually type in the chat box so that quickly we can take that question.
Sir. Sir,
Oh, I think that seems
yes,
to, yeah,
yeah.
yes, Professor
Is
Ramkrishna.
there any regulatory requirements here?
For discovery phase, I don't think so.
Regulatory, yeah.
At least at the discovery part of it, I'm not aware of any regulatory requirements.
Yes.
E. coli is usually the organism where, generally are used,
Yes, fine.
So now I request our session chair, Professor Dr. A. Ramakrishna Savira sir, to felicitate our distinguished speaker, Dr. Sairam Kalpatapu sir.
Yeah. Dr. Sairam Kalapratap, he is presented a very simple way and understanding way, that is the antibody discovery. In this topic, definitely focus on the recent advances, and we can think of the expenditure cost. The cost of the research can be minimized, or we can reduce the time, and we can go with the new antibody designs methods. So it is more informative, and we can work more in this area. Good session here is presented. I take the pleasure of thanking and congratulating Dr. Sairam Kalapatap in this session. And on behalf of the organizing committee, I congratulate Dr. Sairam. Thank you, sir, for your beautiful presentation.
Thank
Now
you.
I request our organizing committee for presenting the momentous.
Yes.
Kritika madam, are you sharing the certificate or what?
Yes,
Hello?
yes, yes. Yes, sir.
Please share.
Yeah. Okay. Now I take the pleasure of thanking the organizing committee, the entire team, and congratulating you for your success, especially Dr. Amulya Ratna Behra for your initiation and motivation for the entire team. Thank you and congratulations. Yes.
Thank you, sir.
Thank you.
Thank you, sir. Now I request our convener, Dean School of Pharmacy, Professor Dr. Amulya Ratna Behra, sir, to kindly felicitate our session chief, Professor Dr. A. Ramakrishna Savaria, sir.
Sir, we are grateful that you have accepted our invitation to be the session chair for this auspicious session by Sri Kalpatabu sir, Dr. Kalpatabu sir. Thank you, sir, for your accepting our invitation to chair this
Thank you.
session. I thank you.
Thank you.
On behalf
Thank you
of
so much,
organizing-
Sairam sir. Personal
Thank you, sir. Sairam sir. Thank you, Sairam sir.
Thank you. Thank you.
And now I'm requesting our organizing committee for presenting the momentous to A. Ramakrishna Sarvaria, sir.
Yes. Receiving it's a great pleasure. Yeah. Thank you.
Thank you, sir.
Thank you, sir.
Thank you. Thank you.
We will meet shortly in the next scientific session with another exciting and informative topic. Till then, goodbye.
Thank you.
Thank you, sir. Siddharth sir. Hello, Siddharth sir.
Yeah.
Sir
Yes,
is
sir.
there,
Yes, sir.
sir.
Please. Actually, we are waiting
Sir
for the
joined.
session chair will come, sir, within a short time. He's actually bit busy with one meeting, so he's coming back immediately. So our session chair, Dr. Prusti has joined, and even I can see Dr. Samir sir. Sir, welcome. Meeting you after a long time, maybe after 15 years.
Yeah, it's really pleasure to meet you again.
Thank you so much, sir. So I request our anchor to please start the session.
So, a very good afternoon to all, respected dignitaries, distinguished guests, invited speakers, and dear participants. I, Gargi Mishra, am truly honored to be your host for Scientific Session 6. It's my great pleasure to extend a warm welcome to each one of you to the DBT-CTEP Government of India sponsored National Conference on AI-driven design and development of new chemical, biological entities and formulations, approaches, challenges, and road ahead, organized by School of Pharmacy, DRIEMS University. Today, we have gathered here to exchange ideas and have insightful discussions in the rapidly evolving field of artificial intelligence-driven drug discovery and development. This conference provides an exceptional platform for exchanging knowledge, exploring ideas, and progressing meaningful collaborations. It is indeed a wonderful opportunity for all of us to expand our perspectives and strengthen our understanding of this transformative domain. So, without any further delay, let's begin this exciting and intellectually stimulating session. Now, it's my privilege to introduce and welcome the session chair for the Scientific Session 6. So, with great pleasure, I invite Professor Dr. Amiyo Kumar Prusti sir to preside over this session. Dr. Prusti is a highly accomplished academic and researcher in pharmaceutical sciences with over 23 years of professional experience. He currently serves as the Dean of the Faculty of Pharmacy at C.V. Raman Global University, Bhubaneswar. A Beatmaster alumnus with exposure to Bath, Mumbai. Professor Prusti's strong expertise lies in bridging conventional pharmacy with advanced biotechnology and artificial intelligence applications. He has made significant contributions to the development of innovative drug delivery systems and nanobiotechnology solutions for healthcare. Dr. Prusti is also actively engaged in AI-driven methodologies for disease diagnosis, with numerous publications in reputed national and international journals and funded projects from the Innovation and Agri-Entrepreneurship Program of RKVY RAFTAAR. He has established himself as a thought leader and researcher in the field. So beyond his research, he has mentored and guided many students in advanced pharmaceutical studies, fostering the next generation of scientists and professionals. So, ladies and gentlemen, with utmost respect, let's welcome our chairperson of the Scientific Session 6, Dr. Amiyo Kumar Prusti sir. I now request our session chair, Dr. Prusty sir, to kindly introduce the distinguished speaker of this scientific session to our delegates. So a very warm welcome to you, sir.
Thank you, Ms. Gargi. So, am I audible?
Yes, sir, completely audible.
Okay, fine. So, first of all, I congratulate to the team for organizing such a nice seminar, national level seminar for 3 days. And I think this is the last day. So, definitely the participants have earned a lot of knowledge on AI-driven drug discovery. And also, I thanks the management and the team, School of Pharmacy, for inviting me to chair the session. So now I welcome Dr. Samir Agarwal. As you know, he earned his PhD in chemistry from Technical University of Dresden, Germany, and postdoc at the Max Planck Institute of Molecular Cell Biology. Then served as a research scientist at Jado Technology, Germany. He obtained master's degree in chemistry from IIT Delhi and was awarded a prestigious DAD fellowship. Dr. Agarwal currently serves as Executive Vice President at Molecule AI in New Delhi. Previously, he held position as Director and Head Medicinal Chemistry at Scondingers in Hyderabad, India, and earlier worked with Zydus also, Life Science. With over 25 years of international experience in scientific research and leadership, Dr. Agarwal has specialized in multidisciplinary new drug discovery. Dr. Samir Agarwal is the co-inventor and project lead of Zydus clinical candidate, Isnoflast, phase 2, a novel oral small molecule inflammasome inhibitory useful in neurodegenerative diseases like Alzheimer's and Parkinson's. With an extensive publication record, he has authored over 50 research publications, patents, invited review articles, perspectives, and book chapters. Dr. Samir Agarwal is a notable figure in the field of medicinal chemistry. His insight on the use of AI in expediting the drug discovery process, particularly in identifying the right molecule through AI-based computer simulations, are invaluable. His work exemplifies the integration of advanced computational techniques in medicinal chemistry, potentially reducing the time and cost of drug development. So let's welcome Dr. Samir Agarwal to share his insight on the topic, AI-driven drug discovery, accelerating innovation in a transformative era. Definitely the need of time, because we know in drug discovery, a lot of time is wasted. And if we can utilize AI properly, then definitely we can reduce the time gap. So without further delay, we'll invite Dr. Samir Agarwal to give his insightful talk. Thank you. Thank you.
Thank you very much, hello, Professor Pushti, for this kind words and a very extensive introduction of mine. Really appreciate it. I also thanks to the Dream University for organizing such an excellent event. And as you really rightly mentioned, this is the need of the time. The application of AI in every field is very much needed. I also really recognize and appreciate the team of this event who is working very hard for this event to organize. I can imagine it is not really an easy job. And also I would like to thank Professor Siddharth Kar, whom I know earlier from my days at IDUS, for giving me this opportunity here. I hope my screen is visible. I try to make it on a screen mode. Just if you can please let me know if my screen is visible already.
Yes, sir, it is absolutely visible.
Perfect. I really appreciate it. Thank you very much. So today I will be giving a talk about AI-driven drug discovery. So accelerating innovation in a transformative era. I represent Molecule AI. So this is an AI-driven drug discovery company, and we have created a platform, an AI-first platform, which helps to design the new drugs. So just to give a glimpse to the audience about the new drug discovery, as you understand, here are the people who are a part of our new drug discovery teams, or in the future, they will going to join the students the part of our new drug discovery teams. So the new drug discovery is normally a very long process. So if you look at the chart, what is depicted on the slide at the moment, so the pre-discovery take itself around 3 to 6 years, and almost all of 5,000 to 10,000 compounds need to be experimentally synthesized and evaluated in the biological assay, which is a very long, tedious job, and it needs large amount of time, resources, energy, and money. And out of those large number of thousands of compounds which have been synthesized, evaluated, out of that, roughly on an estimate around 250, which is also even more, enter to the preclinical where they are evaluated for the efficacy in vivo in the animal models, in the larger species and stuff like that. And finally, out of that, one molecule is really approved by FDA's for a IND submission. So this is an investigational new drug, and this is only an approval for a new compound to enter to the clinical trials, the phase 1, phase 2, and phase 3. And out of every 4-5 molecules which enter to the clinical trials, which really get the FDA approval, is really one. And again, you can see the timeline. It really takes a very long time for that approval, and we need to do a lot of tedious job. So giving a clip about the till IND part or a discovery phase of a drug discovery. So these are the stage gates what are depicted on the slide at the moment. So we do a target to hit, or we can call it as a hit identification, which is mostly the assessment of the physicochemical and in vitro ADME and DMPK properties of a new molecule. So these are the mostly in silico based. We do a lot of effort to identify a new thing, and then we validate that hit experimentally and stuff like that. And the IP is also an important part over there, that the molecule should be novel and they should be IP secure. And then we identify the hit, then we go from a hit to lead. So hit to lead is optimization of the physiochem and the druggable properties. And we do these type of assays into those aspects. And then we go to the lead optimization stage, which consists of optimization of a druggable properties, IVE. So mostly it goes to the in vivo studies into the animals and stuff like that. And finally, the molecule which are showing a good efficacy, it goes finally to the preclinical, we call as a candidate selection, where the dose range, finding safety, tox assessment, interspecies, like from the mice, we go to rat, to the dogs, to the monkeys. So we do the interspecies scaling of these molecules over there. So now coming back to our drug discovery. So drug discovery is a complex multi-parameter optimization problem. So a person like me who is a medicinal chemist and designing a molecule, we design a molecule. So we go to the potency 1. At the same time, we look at the competitor's IP. There can be already an existing ideation. Then our IP may not be that useful. Then we have to look for a potency for the target 2, which is sometimes called as an off-target effect, or it can be also the potency for the target 3. So we can to optimize all these things, it takes a lot of time, and it is running out if you optimize one thing or the other. So what we realize, the chemical space is vast, and can a high performance computing help us design more complete molecules? So this is the biggest challenge, what we try to address onto that. So going to the designing of a drug, as I mentioned in my previous slide, is a very challenging multi-parameter optimization problem. So if you look at the potency, we go with a molecule, we find there's a potency, but the selectivity is missing. We add a small piece of a molecule, we find, okay, now we have a selectivity, but we misses the potency of the molecule. We add something, then we can get, but it is a big, like we have a solubility, bioavailability, clearance, half-life, permeability, drug-drug interactions, synthesizability. So all these parameters need to be optimized in a single molecule. And whenever we do a traditional way and we add a molecule, we can solve one and we are stuck somewhere else. And this is the reason most of the compound even did not enter to the IND phase. They could not able to enter to the clinical trial. And there is a very small amount of success rate is there. This is official figure, but the figures can be even more actually. So what is exactly in the present drug discovery the problem statement lies? So most of the drug discovery program fails to deliver the compounds to the clinic to the inability to identify ligand with the desired properties. So this is the biggest reason. So if we have already the pre-optimized desired property, the possibility of a molecule to progress into the clinic will be high and it will sail through all the stage gates. The stage gate, what I was mentioning in my earlier slide, like hit identification, hit to lead optimization, and finally to the preclinical. So if the properties are well optimized, so it can sail it through with that. So potential solution to accurately predicting drug properties. This is a decade-long challenge now. So now the knowledge-based machine learning, which is often referred also as artificial intelligence, AI. So if the AI can beat humans at chess and go, it can recognize faces in photos, autonomously drive car. So it can also be used to design drugs. So this is the biggest question often all the computer engineer and the coders and the algo people are thinking. And what it requires the deep understanding of physics underlying highly complex molecular interaction. So if we are able to understand the high complex molecular interaction, maybe we can able to design the drug using the machine learning or AI. So what is the vision for the future of our drug discovery? So if you have a very powerful AI and we are using a high speed computing, which can do multiple simulations at a time, and then we can select the best molecule having potency, selectivity, solubility, bioavailability, clearance, half-life, permeability, drug-drug interaction, synthesizability. So we can get and optimize all these features in one single molecule. So we can run over the millions of the molecule and we can select the best molecule which is required for that. So this is what we are saying. If all properties can be accurately calculated, the designing drugs would have been much higher success rate, be much faster, cheaper, and produce much high quality drug-like molecules. So now what is the impact of AI on a drug discovery? So AI is revolutionizing the drug discovery by integration of AI techniques such as deep learning models, machine learning, generative models, neural language processing, NLPs, reinforcement learning, and predictive analysis has significantly improved various aspects of drug discovery. And the key application include the molecule generation, that is the de novo molecule generation, the molecule screening, target identification, binding affinity prediction, which is the most important, whether a new molecule can go and bind to a protein of interest, which is responsible for a disease condition in the humans. Drug repurposing, a very important aspect. If we can really find a new mechanism of action for any approved drug, then it will be, we don't need to go for a clinical trial and that drug can be repurposed for a different disease. And the molecular retrosynthesis. So these are the key features where AI is playing a very significant role actually. So I will just try to explain a little bit to the audience what is the design process here. So what we do actually using the AI and the machine learning, we do a hit identification. So the hit identification is the de novo generation of the molecules. So the millions of molecules, the de novo, which don't have any prior IP sometime existing over there, we can generate those millions of molecules using the de novo generation, AI and machine learning. And then we do the structure determination. This is the structure determination of the protein and sometimes co-crystallize with the ligand. So this is the cryo-EM or the X-ray structure of that. And based on that, co-crystal or the crystal of the protein, what we do a large scale ideation. So the large scale, we do a multiple technologies enable the de novo design. We do a large scale generation of the molecule, and together with that, those generated molecule, we can also do the MPO, the multi-parameter optimization, what I was describing in the earlier slide. And based on that, we can rank order the molecule which are obtained over there, and which only a few molecule out of the millions of the molecule, maybe a single digit molecule, 8, 9, or 10 molecule, go for a synthesis and assay. So based on that, either we can have all the properties optimized and we can directly have, or it helps to generate the SAR into that and it goes. So this is basically a design process using the AI and machine learning. I will give another design process or to tell to the audience in a little bit easier way that how does it work. So we can simply do a de novo generation of the molecule in the order of a 1 billion. It's not million, it's billion. So we can do just with the speed of the computer, and we have got a lot of GPUs these days, which are ultra supercomputer equivalent type of a GPU, which help to run the simulation very fast and which enables the de novo generation in the order of a billion molecules. And then out of that, you can just choose the 1,000 molecules, compute the property using AI, and on the day one, you can generate the molecule, train the machine learning models into that, and based on these enriched molecule, you can again score 1 billion more molecule, which are looking good, and the properties are already optimized with those molecules. And now out of those 1 billion molecule, which are already generated, which is already having drug-like properties and the potency of a binding affinity, you can select the few thousand compound which are already enriched very highly. These are the good molecules. Again, you compute the properties using AI and machine learning, and then out of that, you just pick up a few compound, as I said, in a single. So out of a 1 billion generated here, we generated one more billion compound, which are enriched compound, and then we are choosing only very few compounds. So you can imagine the power of a computing, that we are really choosing the molecule where all the properties have been already optimized. So when it gets a binding affinity hitting, so it can sail through all the stage gates of a drug discovery because the properties have been previously optimized. And out of the 10 molecules synthesized, the possibility that the 8 molecule will advance the program very quickly. So this is the accuracy, the combining accuracy with the speed of AI enables ultra large exploration. So we can really ultra largely chemical space could be explored. So now I'm just coming to apply the deep learning to the molecular modeling. So the molecular modeling, the deep learning models are used to simulate the complex interaction between molecules and providing into the drug target binding or binding affinity. The protein structure prediction. The AI can predict the three-dimensional structure of protein, enabling the design of drugs that are specific on the protein side. So we can have a new molecule which can just directly bind to the protein of interest, which is responsible for a disease condition into the humans. And these insights can accelerate the discovery of new drugs. So now what is the AI technology? So AI technology, the machine learning identifies the pattern in the large data set. So the application mainly is the target identification, virtual compound screening, and predicting drug target interactions. The deep learning uses neural network to complex data representation, and it is applied for image analysis, molecular structure prediction, and novel designs. The generative model uses the GANs and VANs to design novel molecular structure, what I was talking about, the de novo designs with the desired properties. NLPs, the natural language processing, extract insight from literature, patent, and databases. A huge amount of literature, patent, and databases, which no human can really, up to a certain extent, can do that. And it can extract those data from the humongous amount of data which is present, and it can identify relation between genes, proteins, and the diseases, which is very efficient in doing that process. The reinforcement learning optimizes the drug designing by generating and evaluating compounds for efficacy and safety. And finally, predictive analytics. So the predictive analytics forecast the admittal property and the efficacy to aid the drug discovery. So the AI techniques further, the molecule generation, we have already talked. So the generative models create a diverse pool of novel drug molecules. The molecule screening, this involves identify the most promising candidates based on the predicted properties. AI can predict the drug compounds admit properties, allowing research to optimize the lead structures. In addition to that, the AI plays an important role in target identification. So AI aids in pinpointing the potential biological target for drug action by mining gene expression and mutation data. AI model can predict which proteins or genes are likely to interact beneficially with drug candidates. So this is a very important and efficient way how AI is playing a role into that. So the further going to the key applications of the AI, the binding affinity prediction. So this is the foremost thing for any drug discovery program, that any novel molecule should go and bind to the protein of interest. So this focus on the interaction strength between the drug candidate and its target protein. And the performance of AI model in this area can be significantly affected by distribution of training data. Drug repurposing, I've already discussed about that. AI facilitates the identification of new uses of existing drugs and the molecular retrosynthesis, which is a very important, because the AI is generating the molecules. Sometimes it looks very strange to even to the very skillful organic color synthesis, but they may be very efficient at a disease level. So the deconstruction of the molecule to identify synthetic pathway, that also AI can help, and that will be helpful for synthesizing those novel molecules which are predicted by AI. So as I mentioned, the binding affinity is the first starting point for any drug discovery program. Any novel molecule has no value unless and until it goes and binds to the protein of interest. So the molecular docking, this technique predicts the preferred orientation of a molecule when bind to a target protein, providing insights into strength of interaction. The machine learning, the recent advances have seen that application of various machine learning algorithms to predict the binding affinity. So we also have an in-house program where we can predict the binding affinity very precisely onto that, and just using without any long simulations required. So on a very high throughput scale, we can run a very quick AI-driven platform, and it gives a prediction about the uh, binding affinity to the protein. Data shows the accuracy of the binding affinity uh, heavily relies on the quality of data used for these training models. I will now just touch that how the AI has impacted the drug discovery and what are the recent advances onto that. So these are some of the notable AI-driven drug discoveries, and these are the success stories, rather I will tell. The in silico medicine is leading on to that. It was a start a few years back, but it has really driven very quickly. So in silico's drug candidate called INS 018055 for the treatment of idiopathic pulmonary fibrosis has become the first entirely AI and AI designed drug to enter to the phase 2, representing an important milestone for the industry. So this is the first entirely AI driven molecule which has entered to the clinic, and now it has entered to the phase 2. Excentia is one more company. It has the two molecule which is already into the clinic for immuno-oncology and as an immunomodulator drug candidate. They both are into the phase 1. There is another company called Becnovelet, which has a compound for the ulcerative colitis. So this is for that compound. In addition, the Nimbus and Takeda deal, the very famous Nimbus has sold for the $4 billion to Takeda, the allosteric TYK2 inhibitor. So this is one of the very key in the AI or in silico based deal has happened, and such a large amount has been received as a upfront payment by this company. The Alphabet's isomorphic lab is doing an amazing job. It has a tie-up with the Lilly's and Novartis. So Alphabet Isomorphic Lab has formed a collaboration with Eli Lilly and Novartis, but potentially worth up to 3 billion plus seller royalty for the focus on a small molecule drug discovery. Schrödinger, where I was working earlier, they have a big collaboration with the Bristol Myers Squibb, and this collaboration focus on discovering small molecules for multiple targets. And Schrödinger has received the $55 million as an upfront payment for that. And there are some of the smaller players which are also doing a very good job. Valo has signed a deal with a Pfizer. Verge Genomics has a deal with a Lilly for a neurodegenerative diseases. So I'll just put a case study how this in silico AI machine learning things are so efficient. So I take a case study of the Malvern program from Julius Schrödinger. So the Schrödinger has generated almost 8.2 billion molecules code with the machine learning. And as I was telling, how does this design process goes? The de novo, and they train the model, and they go further, and they generate the more molecule. Then they have the enrich molecules, and finally they select the molecule with optimized properties, and which has the highest binding affinity and stuff like that. So out of the 8.2 billion molecules, they have modeled and scored with the physics around 12,000 molecules. And out of that 12,000 molecules, they have only taken 226 molecules, to be very precise, to their wet lab. Only 226 226 molecules have been synthesized and evaluated in the assays. And out of that 200 molecules, they have already found a development candidate which entered to the phase 1. And all this process happened less than a year. So you can imagine the how efficient process right from a concept to the clinic with this case study, how the machine learning can do efficiently the things to bring the compounds to the clinic. I'm going further with a notable AI-driven drug discoveries. Atomwise is another very strong company. Is a AI-powered pharmaceutical company that uses deep learning to discover new drugs. Their AtomNet technology has helped identifying new drug candidates for a wide range of diseases, including cancer, infectious diseases, neurological disorder. And AtomNet searches a chemical library of over 15 quadrillion compounds to find its hit. So you can imagine now I was talking about a few 100,000, then we went to million, then we went to billion. Now it's not even a trillion, it's a quadrillion compounds. So AtomNet has, or Atomwise as a company, has devised that all the molecules, whatever one can imagine, they can take out those molecules to find the hit which really has binds to the protein of interest and has all the feature which can make it to the drug-like. Recursion Pharma is another very amazing company which focus on indications with a high unmet need, including oncology, rare diseases, and infectious diseases. They have a molecule called RAC3565. This is an oncology drug that shows tumor growth in inhibition in a xenograft models. Another very new startup, or it was a new startup, Awesome Biodiscovery. This is an absolutely an amazing company. They have an AI-powered platform called as a Alscreen platform. This is a very famous Alscreen platform. And for what they do actually, they integrated their tools to predict the unresolved 3D structures. So normally everybody look for a cryo-EM or the X-ray crystal structure of a protein, if not with a ligand, at least of the protein. So what they are doing, what they have integrated their Alscreen platform for the unresolved 3D structure where there's no real-time experimental cryo-EM or X-ray structures are available at all. And they incorporated the conformational dynamics and identify the potential binding sites, paving the way for the hit identification phase. So Awesome is doing in that way an amazing job. And I will finally come to the DeepMind. We cannot, if we talk about AI, we have to talk about the DeepMind because they are the one who revolutionize structure biology by predicting the protein structure with high accuracy, widely used by researchers worldwide. So to give a glimpse to the audience what type of impact AI or machine learning is doing, so you can imagine, so these are the deals with the science company, life science companies, and by 2024, that more than $200 million deals has been done. And like all these big companies, Lilly's, Novartis, Bristol Myers Squibb, Boehringer Ingelheim, Merck, Novartis, they are tying up with the companies like Isomorphic Lab and all these companies over there. And most of these deals are for our drug discovery deals. So they would really want to do a serious drug discovery from a concept to a clinic. And you can imagine that the amount of the money involved is huge, and these are the deals leading to a drug design. So they would like to identify the hit basically, and they are the few companies or the few deals which are about the target identification. So I just want to give the glimpse that impact of AI globally and with the big companies as well is very high, and they are investing huge amount of money into that. So future is here. And this is another life cycle chart. And all these AI companies, where they stand, where they're using the AI right from the identification to the drug discovery, development, and post-approval. So you can imagine everywhere there is a high impact of AI ongoing with that. Here, I just want to tell this is a 25 leading AI company to watch in 2025, and this is by a biopharma survey they have made, and this is where I'm associated with a molecular AI. So this is one of the most search and most impactful startup or a AI platform company, which is creating an impact in a drug discovery. So the AI revolutionized protein structure prediction. So DeepMind's AlphaFold has revolutionized structure biology. It predicts the protein structure with accuracy. So we don't need any cryo-EM or X-ray structures. It can just predict that. So the breakthrough in the protein prediction, the deep learning algorithms are used for a protein folding prediction. In 2021, AlphaFold predicted the structures for more than 330,000 proteins, and it has created a global impact. It is widely used by researchers and companies worldwide. And the 24 Nobel Prize, of course, went to the AI in chemistry, where the David Baker, Demis Habbassis, and John Jumper for their discovery on the AlphaFold has got. So this is one of the amazing AI driven work which is done and which is really crucial for our drug discovery. So in the clinical trials as well, the drug discovery is doing a very impactful job. So for the patient recruitment, if some of you are involved into the clinical trial, sometime to identify or to find the right patients for a right drug is sometimes very tricky. So AI algorithm can analyze patient data and identify, recruit the most suitable participants for the clinical trials, ensuring that the safety, the studies are powered with the right patient for optimal results. In addition, the AI can also optimize the clinical trial designs based on the historical data and predictive models. And of course, the algorithm can analyze the vast amount of data which is generated during the clinical trial and very efficiently, which is sometimes might be missed by humans. I just want to touch a little bit very quickly that what are the challenges associated with the AI-driven predictions. So out of the distribution, models trained on the specific data set may perform poorly when faced with a novel compound that differs significantly from the training set. It may happen sometime, but the research is going to get where we can predict the binding affinity at least up to very precisely. Identifying the most relevant feature that influences binding affinity remains a complex task, requiring domain expertise and multiple data testing. And finally, the inconsistent or incomplete data can lead to inaccurate predictions, necessitating robust data curation practices to ensure reliable results. So these are the challenges. Of course, every new technology has the challenges over there. I will quickly do a comparison of the drug discovery, evaluating traditional versus AI approaches. So if you look at the efficiency, the traditional method may take around 10 to 15 years to develop a new drug, while AI can reduce the time for 1 to 3 years. Traditional method, of course, in terms of cost, was costing on an average to the order of $2.6 billion, and it has cut down to less than $1 billion. Accuracy, the traditional method have been lower precision in identifying viable candidates. AI technique can improve the accuracy up to 70%. Innovation, the traditional method rely on established technique. The AI method leverages advanced algorithm and data. So innovation, the de novo generation of the molecule, what I mentioned, are mostly novel molecules and no infringement to the prior art. These are the completely novel molecules which are being identified by our AI-driven method. Success rate of our traditional method is around 10 to 15%. In AI-driven method, can enhance the success rate to 20 to 30%. And this success rate can even further increase when we have a more powerful and accurately predicting tools. So this is an evolving field, and every day we have a lot of new innovations in this field, and which is helping to predict the molecules in a very precise way. So the future of the drug discovery lies in the drug discovery efficiency and the personalized therapies, drug repurposing, drug prevention and treatment. AI can also predict and prevent disease by identifying individual at risk. So suggesting the preventive measures, improving overall health and the well-being. So I will just give a quick that how it is going to pan out. The traditional method provides a solid foundation. These methods have historically led significant breakthrough in finding new drugs. AI introduces efficiency and precision. AI enhances the drug discovery process, making it faster and more cost effective. Hybrid approach will foster innovation. Combining the traditional drug discovery and the AI method will drive the advancement in the therapeutic development even more efficiently. AI integration will enhance therapeutic development. As AI involved, it will create a new pathways for a rapid development of the new drugs. So at the end, we must tell the AI should be used as a tool to augment human expertise. This is not at all a recommendation to replace it at all. It is only to augment the human expertise onto that. And the Nobel Prize winner from the AlphaFold, Demis Habbis, has told, I have always thought if we could build an AI in a right way, this could be ultimate tool to scientists to help explore the universe around us. So I just quickly say I was working at Zydus at a program like a NLRP3, which also I was working on a programs like a GPR40, which has been advanced to the very advanced select clinical preclinical trials. I was very leading a program on a TGR5, and the results has been published into the multiple papers onto that. We had a very efficacious compound for a TGR5. And for a NLRP3, the program has been progressed to IND and finally to the phase 1, and now it has entered to the phase 2, and now it has reached the INN name, and now it is called as a Flask to that. With that, I really thanks to all the audience and again to the organizations for having me with these quotes, and I will be happy to be approached at any time. These are my coordinates. Thank you very much.
So, thank you so much, sir, for the wonderful presentation on AI-driven drug discovery, accelerating innovation in a transformative era. So now we will move on to the question and answer session. Dear participants, you all are requested to post your questions or queries in the chat box. And I also request our session chair, Dr. Prusti sir, to kindly take over the session. Over to you, sir, for the question answer session.
Yes, participant, you can put your question on the chat box.
I have a small doubt, sir. Siddharth this side, Tamir sir. Sir, already, am I audible?
Yeah, audible.
Yes, sir.
Sir, already you have seen a transformative research since last 25 years, from very orthodox research, conventional research to now the AI integration in medicinal chemistry. So how do you see it? How difficult it is for a traditional orthodox medicinal chemist to become AI-driven medicinal chemist? So what is your perspective on this?
Dr. Agarwal, are you there?
Excuse me, sir.
Maybe
Agrawal, sir.
some network issues.
Yeah, might be.
Yes, sir. I think there is some network issues.
Yeah,
So,
he may
sir,
rejoin.
Yes, sir. Wait for a few minutes. So participants kindly wait for a few minutes due to some technical issues from Dr. Agrawal sir. We're facing there some issues. So we will Agrawal sir, kindly join again.
Just wait, just wait. Just wait a minute.
I'm sorry, I think something happened with my net. I'm sorry about that.
Oh,
Some glitches. So you were asking something, I'm sorry.
Actually, sir, my question is very conventional one because you have seen a change, transformative era from very conventional medicinal chemistry
Yes,
to
exactly.
AI-integrated medicinal chemistry. So,
Mhm.
for a very conventional medicinal chemist, how it is difficult to become AI-driven one? What are the transformations he needs to do?
No, actually, this is a very nice question. I really appreciate for that question. And most of us come with a traditional drug discovery background because AI is very new, I would say to that. But I think the person having a strong traditional drug discovery, for them to understand AI and to apply the AI and use it to the right human expertise augmented on the top of the AI is the most important aspect actually. So the people having a vast traditional drug discovery, for them to use their traditional knowledge and augment it with the AI is the need of the time. Only AI, it is very difficult that the computer will give a molecule and it will become actually a drug. So you have to use that human expertise together with the AI, because AI can really help you to identify some of the new hits, because sometimes it is very difficult and we all come with a traditional background. So for any drug discovery target to identify a hit experimentally, you have to go with the techniques like HTS and DEL, and which is very expensive actually. So we can take the molecule from there and using our experience, we can sail it through. And if all the properties are well optimized, you can directly from a hit identification, hit to lead and lead optimize, you can sail it through very quickly. And this is the essence, that you can really reduce the time, money, energy, resources in a drug discovery program with that.
Right. Correct. Thank you, sir. Okay. Okay, over to session chair. Sir, you please unmute yourself.
okay. Participant, do you have any questions? Please put it on the chat box. I think there is no question. No, some question is there coming up. Yes, Aditi has a question. Sir, this is a question in the chat box. A second year B.Pharm student. I understand that AI is helping a lot in drug discovery and in making research faster, easier, and more effective. But at the same time, we also see in many IT companies, the employees have lost their job because of AI automation. Since India has a large population, don't you think AI might create similar challenges in the pharma sector in future, especially for middle class people? And finally, if we students want to build a good career in pharmacy with AI, what extra skills or subjects should we learn from now so that we say relevant and get better opportunities? Thank you, sir. Agarwal, sir?
Yeah.
Sir, please.
So,
This is a
so
nice question from student point of view.
Yeah. So, Titi, I really appreciate for your question and your concern and your aspirations to grow in your life. It is really appreciated for that. As I mentioned, Titi, in my talk, and overall, this is my strong belief that AI is not replacing human by any chance at all. That is only augmenting it. So the more skillful you are, whatever job you are doing, you will be more efficient in doing your job. AI is helping you to make it more efficient in doing the job. So as a drug discovery researcher, if I was using a traditional techniques, or for example, on a literature or a patent, I may take a day to dig out the information. Now using in our few minutes, all the literature, vast amount of data and the literature, I can take and I can analyze, and I can take more better and efficient decisions based on that. So AI is not at all losing any job at all. It is creating a job and making you more efficient.
Yes, sir.
And to
Definitely.
learn AI and to IT, this thing and application of AI is always really useful.
Sir, another question also, she asked what training the pharmacy students should undertake so that they can have a good career in
Right. So I think a little bit coding, if you know how we can code for designing, or you can take a internship in any of the AI type of a company. So if you're working for few months as an intern, you will come to know how exactly they are actually working. So to know and to being aware and simply curious. Einstein has said, I am simply curious. So if you are only curious to know something new,
Definitely.
nobody can stop you actually. So I would really suggest you, Aditi, you are very young, you are just still a second year student. Just still reach out to a lot of new companies are there. They are doing phenomenally excellent job and they need a lot of interns. So if you can join them as a few months for an internship, that will give you insight and that will also align with your interests and your aspirations. That you are really suitable, you are finding it good or not, you find interesting. If not, you can see something else. So that is a good way to do it.
Right. Thank you, sir. Thank you. I don't think no more questions. So, Gargi, over to you.
Yes, sir. Thank you so much, sir, for sharing such valuable insights with us all. So I now humbly request our session chair, Dr. Amiyo Kumar Prusti, sir, to felicitate our distinguished speaker, Dr. Saumil Agrawal, sir.
Okay. So, thank you, Dr. Agarwal, for giving such a insightful talk. And from your talk, what I come to learn is that by using this docking, generative AI or machine learning, we can reduce the time as well as the cost. Simultaneously, we can increase the efficiency of the drugs, and definitely it will be a great help for the society for development of new drugs for incurable diseases.
Exactly.
So thank you. And
Thank you.
definitely we are going to share you. Okay. So now this is for your insightful talk
Thank you
and
very
a
much.
love of token
Thank you.
honor. So
Thank
please
you.
accept on behalf of the team,
Accepted.
organizing team, as well as Dreams University, as well as from my side. So
Thank
thank
you
you.
very much.
Thank you, sir.
Thank you. Really appreciate it.
Thank you very much, sir. So आज we come to the end. Before that, now I request our beloved Dean Research, Dreams University, Professor Dr. Amulya Ratna Behera, sir, to felicitate our session chair, Dr. Amiyo Kumar Prusti, sir.
Thank you, Amiya sir, for your valuable time to chair the session through Zoom platform. Thank you, sir. Thank you, Samir sir, for
Thank you.
enlightening the participant regarding
Thank you very
this.
much.
Thank you.
Thank you, Professor Amulya, for giving
Thank
such
you.
a nice chance.
Yeah, I really appreciate
Thank you, sir.
all of you to organize this event. This is really very too much work to organize such events.
Thank you, sir.
And also a special thank to coordinator, Dr. Siddharth Kaur.
Yeah,
Thank you, sir.
really
Thank you, sir.
thanks to Dr. Siddharth.
And all the team. Okay. Thank you.
Thank you. Thank you,
Good
sir.
Thank you
day.
very
Good day.
much. Okay, bye-bye. Thank you.
Okay, thank you very much to all distinguished speakers and all participants. As we come to the close of this scientific session 6, I now extend my heartfelt gratitude to our esteemed speaker and session chair for sharing their profound knowledge and expertise. I also sincerely appreciate all the participants for their active engagement and thoughtful contributions. It has been my pleasure hosting this session, and I hope each one of you is leaving with deeper insights and fresh perspectives on our focus topic. So by looking forward to having you all back with us after a short tea break as we continue with the scientific session 7, So thank you once again for your patience and support. Have a great day.
I think Professor Anuradha Mishra, madam, are you present?
Sir?
Professor
Certainly, sir.
Anuradha Mishra, madam.
Yes, yeah. Namaskar and good afternoon,
Namaskar,
sir.
madam.
I am here, very much here.
Yeah. How are you, madam?
I am good. Congratulations for organizing such a nice conference. I am here
Thank you,
in the last
madam.
session itself.
Nice to see you after a long period of time, maybe so around 20 years. So
Yes, yes.
and very happy to see you in such a good position.
My
Okay,
best
fine.
wishes
So
are there for successful completion of this DBT sponsored national conference. I am going through your this topic is really very nice. What you have
yeah.
selected, that is the real challenge nowadays. And in our next year onwards, syllabus is also modifying, including
Yeah, that's
AI.
true. Very true.
So
They are
such kind of
incorporating
conference is really required.
it's essential. Okay, so let me see whether Dr. Rajneesh has joined. Dr. Rajneesh from BH. Dr. Rajneesh, are you there? Okay, actually, Dr. Rajneesh is joining. He has some technical issues, so he'll be joining very shortly. So please bear with us for another 1 or 2 minutes.
Hello everyone.
Yes, sir. Great. You are visible and audible also. Perfect.
Yeah, just a minute. I just open the presentation.
Good afternoon, ladies and gentlemen, esteemed guests and conference participant. Welcome to DBT-CTEP Government of India sponsored National Conference on AI-driven design and development of new chemical and biological entities and formulations, approaches, challenges and road ahead, organized by School of Pharmacy, DRIEMS University. I am Ms. Rosalind Moharana, your host for the Scientific Session 7. It is indeed a golden opportunity for each one of us to enrich and enlighten ourselves. Without further ado, let's dive into our promising schedule. Now it's the time to welcome the session chair for the Scientific Session 7. It is my great pleasure to introduce our session chair of the session, Professor Dr. Anuradha Mishra Madam. Professor Mishra currently serves as the Professor of Pharmacology and Director of the Amity Institute of Pharmacy, Lucknow. She is a goal-oriented professional with over 19 years of super-headed academic and research experience in the field of pharmacology. Her research specialties include the pharmacological screening of plant extracts, bioactives, and synthetic drugs, as well as improving their deliveries through the nano delivery system, targeting neurodegenerative disorder, metabolic disorders, and liver cancer. Her research has received recognition from the UPCST in the form of an extramural grant. Dr. Mishra has supervised 11 PhD students and 21 postgraduate theses, and she is currently mentoring 5 PhD candidates and 1 postgraduate scholars. She has published over 90 research and review articles in national and international journals of repute, many with high impact factors more than 5. H-index 17 in addition to authoring 3 books, contributing to 7 book chapters and holding 5 national and international patents. Ladies and gentlemen, please welcome 2 eminent personalities. First, our chairperson of the Scientific Session 7, Professor Dr. Anuradha Mishra Madam, and our distinguished speaker of the Scientific Session, Dr. Rajneesh Kumar, sir. Sir is a renowned expert of molecular modeling and drug design. He will deliver a presentation on AI in drug discovery, opportunities and challenges. I request our session chair, Professor Dr. Anuradha Mishra Madam, to kindly introduce our distinguished speaker to our delegates.
Thank you, Ms. Rosalina. I believe that I am audible to all of you.
Yes, ma'am, you are audible.
First of all, I extend my heartfelt thanks to giving me this opportunity to be a part of this national conference. Cordially, I invite Dr. Rajneesh Kumar to be a part of this conference and giving your talk on the specific topic. Dr. Rajneesh Kumar, an associate professor in the Department of Pharmaceutical Engineering and Technology at IIT Varanasi, is a recognized expert in AI-enabled drug discovery with advanced training and notable achievements in neurodegenerative disease therapeutics, particularly Alzheimer's and Parkinson's disease. He completed his postdoctoral training at the prestigious Karolinska Institute, focusing the preclinical discovery of lead candidates for neurodegenerative disorders. Further enriching his expertise, he conducted research at the Institute of Protein Research, Osaka University, where he worked on a predictive modeling of ADMET parameters. Dr. Kumar's research integrates predictive modeling, virtual screening, and high-performance computing to expedite the discovery and optimization of therapeutic leads, with a strong emphasis on developing small molecules that inhibit protein aggregations, which is a key factor in neurodegenerative pathology. His innovative approach extends to diagnostic as well. He is working on PET and SPECT tracer developments for choline acetyl transferase imaging aimed at detecting early biomarkers for Alzheimer's disease. With an extensive publications record and funding from both national and international agencies, Dr. Kumar's contributions have been recognized with numerous accolades, including the Young Scientist Award 2023 from the Bioinformatics and Drug Discovery Society at CSBDD 2024, SRM Institute of Science and Technology, Tamil Nadu, and the BP Dr. Young Investigator Award presented at the 15th International Symposium on Cholinergic Mechanism in Marseille, France. Dr. Kumar's advisory experience spans academic and industry platforms where he consults on AI applications in drug discovery. As an advisor, he brings a deep interdisciplinary understanding of medicinal chemistry, computational drug discovery, and AI driving advancements across the pharmaceutical fields. Since September 2024, Dr. Kumar has also served as an editorial board member for Scientific Reports, Nature, Springer. So with that, I would like to inform here the topic on which Dr. Kumar is going to brief. It is briefing on the lead compounds, the identification of optimization of lead compounds, as well as the computational approaches, generative AI, how you can utilize them for synthesis of new molecule or identification of lead compounds. So however, despite these opportunities, there are several challenges which can persist: the reliability and interpretability of the generative models. It remains in critical concerns, as do biases in training data sets and the difficulty ensuring in synthesis as well as feasibility of generated compounds. So bridging the gap between in silico predictions and experimental validations is another bottleneck that must be addressed. Ethical considerations, data governance, and regulatory acceptance also plays a pivotal role in shaping the real-world impacts. So with that, without further delay, I would like to request, sir, you please proceed.
Thank you so much, madam, for a kind introduction, and many thanks to Professor Siddharth for the kind invitation as well, and DRIEMS University. Although I was, I'm also conducting 1 week hands-on workshop, and I had to take another, I had to take a hands-on session now, but then I got a call from Professor Siddharth, and he said that your talk is scheduled now. So I just, I'm in the another room, the hands-on session is going on and taken care by my research team, and then I'm delivering this talk. So I think that's the duty of academic faculties, that we have to, continue sharing knowledge wherever we can. And thanks to Rosaline as well for the kind introduction to the session chair. So so today we will talk about, I will give you a brief idea of, like how the AI is enabling drug discovery and development. So if you have any question in between, you can just stop and ask me, or we can take the questions in the end as well, right? So I've been fortunate to work at this institute called Karolinska Institute, where, the Nobel Prize in Physiology and Medicine is being decided actually. So the Nobel Prize Committee at Karolinska Institute, they sit in this Nobel Forum. This is a building situated in Karolinska Institute, Solna campus. Then they sit here in this building, and then they decide to whom they shall give the Nobel Prize in Physiology and Medicine every year. Does anyone know who has got the Nobel Prize in Physiology and Medicine this year? No? I try to keep my, talk a little bit interactive, asking a lot of questions. So just excuse me if you feel like, a little bit irritated. Okay. And then the Center of Alzheimer Research where I was working, it had multiple, the Karolinska Institute had multiple campuses, three campuses. And then the Center of Alzheimer Research where I was working, so multiple people were involved in different kind of research like structural biology, molecular imaging, diagnostic imaging, and epidemiological studies. And it is a center of, advanced study where the world leading researchers are contributing to the field of not only drug development but understanding the basic biology of Alzheimer's disease. So coming to the key thing, that is what is a drug actually. So if we look at the simple definition, we have been using drugs or substances to get rid of the diseases or get rid of the problems long ago. For centuries we have been using them. So if we look at the simple sentence which says that a substance that is used to prevent or cure a disease or ailment or to alleviate its symptom, so it has multiple definitions hidden in it. So it can be a substance which is preventing, means which is used as a prophylactic treatment, which is not treating the disease but it is preventing the disease. That is called as prophylactic therapy, where you are using like all those multivitamins or iron capsules or folic acid so that you do not get sick. Okay. Or you use these drug to cure a disease, like you use antibiotic to kill the bacteria which is there in your body and then you get rid of it. Or you use them for alleviating the symptoms, like we use the paracetamol, and that paracetamol is not curing the disease, but it is just helping you to get rid of the symptoms like the fever or the pain, etc. And the discovery and development is really complicated stuff, where it starts with the target selection first, and then in target selection you have to look at whether the target is validated, and then whether there is a robust assay system available, or whether the structural information is available, or what are those lead inhibitors. And once you have an identified target, then you goes to design the inhibitors, and you design inhibitors by using different techniques, like either through high throughput screen, where you screen thousands or millions of compounds in physical assays. Okay. Where you can also use you can identify these inhibitors from natural products or traditional libraries like Chinese medicine, Unani medicine, Ayurveda. And then once you have identified an inhibitor, so it may be a potent one or may not be a potent one. So you have to optimize it actually. And that step is called as validation, or we can say like lead optimization, or we can say hit to lead and lead optimization, typical medicinal chemistry approach, where you convert these inhibitors into validated hits and then you convert them into leads. And then here you have to do a lot of studies. You have to screen them in vitro assays, you have to screen them in animal models, you have to perform the pharmacokinetic analysis, you have to optimize the efficacy and quality of these pharmaceutical substances. So, and it's an iterative cycle actually. So you do some changes and then you test them. If you are happy, you keep them. If you are not, then you go back to design part, and then you design new inhibitors and synthesize new inhibitors. So this is called as design, make, test, and analyze, DMTA cycle. Design, make, test, analyze. Okay. And once you are happy with all those properties, then you get a good potency, you get good efficacy, you get good pharmacokinetic, you get like good toxicological parameters. So you finally get a drug candidate which can be moved into the development state, where in the but if you look at the success rate, the overall success rate is very low, like 2 to 5%, costing a lot of money, millions of US dollars, and taking a lot of time. And then when the drug candidate moves into the development phase, so in the development also you have to do a lot of studies, like you starts with the GLP, preclinical safety studies. So GLP studies, preclinical safety GLP studies, where you have to perform mutagenicity, detailed pharmacology, acute toxicity, pharmacokinetics and ADME, subacute repeat dose toxicity, and all these you have to do in animals actually. Okay, and once your compound shows promising data in this preclinical safety studies, you move to the clinical trial, where you in phase 1 your compound is tested, evaluated in healthy humans, and then your compound is tested in disease humans, and then further in disease humans to see whether the compound is acting or not. And finally, once every outcome is affirmative or positive, so then you file for registration, that is called as investigational new drug application, INDA. And then the regulatory agencies like FDA or EMA, European Medical Agency, or CDSCO in India, so they they decide whether to give permission to you to manufacture and sell this drug or not. And then it is available in the market, and then patients can be treated with that drug. So you can see the development phase is also quite rough. The success rate is 5 to 10%, 20%, and then costing a lot of millions of US dollar and taking a lot of time. So overall, if you combine drug discovery and development phases, so it costs around $1 billion US, taking 8 to 10 years, and the success rate is barely 1 to 2% actually. And that is where it becomes a dead end actually. Like you invest a lot of money, in the end, if you're not getting any drug approved, so then you lose, the companies are losing a lot of money. So, and to emphasize on it a little bit, I can give you this example of a successful development of blockbuster drug, omeprazole. So the company AstraZeneca started to explore anti-secretory compounds back in 1972. So they identified the CMN131 compound, which was anti-secretory, means it could inhibit the secretion of acid in the stomach. But what happened was, when they identified this compound, they found that this compound was showing liver toxicity. So they were not able to use it actually, because it is toxic and you cannot take it further. So you cannot use it. And then they changed it into another compound. They made new compound out of this compound, and that was H12426. However, when they synthesized this compound and tested this compound, they again realized that this compound was, having patent issues, because this compound was patented by another pharmaceutical company, so that they cannot use it, because either they have to buy the patent or they have to identify a new compound. And then they further modified this compound, which was a derivative of this H124 timoperazole in 1974. However, this compound was also having severe side effect of inhibiting the iodine uptake, so they cannot take it further and they cannot use it for further development. And then they further converted this molecule into picoperazole, so where the compound was again showing severe side effects of necrotizing vasculitis, so it was also useless. So you can see, like, the challenges where they started from one compound, and each time when they were reaching at the the drug candidate level, so they were facing these issues in GLP. Actually, in the development they were facing issues, and then they have to go back to lead optimization and identify new compounds. And finally, in 1979, so they identified this omeprazole as a potent non-toxic inhibitor of proton pump in stomach, and which is then became a blockbuster drug and used almost by every person in this world. And by the way, blockbuster drug means, like, it's like a blockbuster movie where the annual sale of a drug is more than $1 billion US. Okay, and $1 billion US, you can just imagine, like, if you convert it into crore, so it will be more than I think around 1,000 crore it will be actually. So 1,000 crore sale is coming from only one single drug, and that can change the economy of countries. Actually, in those smaller countries, it can change the economy of those countries. Right. So then they developed this esomeprazole. So this example was just to give you a glimpse of how much challenging, how difficult the drug discovery process is. Okay, now coming to the machine learning part. So if I ask you the question, what will be the next number? Can anyone tell me? If you don't want to speak, you can just type it in the chat box. I can see it from there. But I want your answer actually. What is the next number? Am I audible? No?
Yes, sir.
Then participants, I can see there are 77 participants and no one is responding, means it's not good actually.
Uh, I request all the participants, please write your answers in the chat box.
No, maybe they are sleeping actually. Okay, no worries. So you can say, for example, if I ask what will be the next number, so the next number, what will be the next number now? Tell me.
Excuse me, sir. I think it will be 10.
Yes, yes. So Dipti, Himanshu, Soumya, Ajay, Subrashri, Bhuvneshwari. So they all say 10, right? And that is the correct answer, right? So it is 10. But can any one of you tell me answer to this question? Which of the following can cross the blood-brain barrier? Can you tell me? Any one of you? Which one of the following can cross the blood-brain barrier? Or do you think we can answer this question or not? Yes or no? No, right? Why? What is the reason we are not able to answer this question? We were very fast and we were very accurate while answering the earlier question. Okay, so the reason is why we are not able to answer this question, because here in this question we were able to identify the pattern, right? We could realize that every time we are adding 2 into the previous number and we are getting the 10. But in this case, we are not able to identify the pattern, or the data is, not interpretable by us. Okay. And that is where the machine learning or AI can come into picture. Sorry. So that is where the AI or machine learning can come into the picture. So what AI can help here with? So AI can identify the pattern, like how these molecules, what kind of pattern these molecules have. Sorry. So the pattern can be identified computers, because the first thing is that they are fast, okay, and they are very good at computing or identifying the pattern. So if you look at the AI, I'm sure that you have already seen this picture. So AI is, this big circle where everything comes under AI, like the knowledge bases. And then a sub part of AI is machine learning, where you use the logistic regression, for example. And then you have the representation learning, where you use the shallow autoencoders. And then you have the deep learning, where you use the multi-level perceptrons, where you don't have to explicitly tell the computer what to do. It can automatically identify the pattern. It can understand the data, identify the pattern, and it can make a decision on the basis of that. Okay. So basically what we are doing with machine learning is we have the big enough archive previous observation or past data, which is called as known data. And then we try to identify the relation between this data, previous data, and the outcome actually. And then we identify the unknown data. We predict the unknown data. It is like the weather forecasting system, what it does is it takes the previous historical data of weather by using different sensors, temperature, vapor pressure, atmospheric pressure. And then after taking all this data, it puts it into a mathematical model and try to see relation between, this data. So once it gets that relation, so then it can predict, that whether what will be going to be the future data actually. And that is how we can use it for in the case of drug discovery. So this is where we can use the AI in drug discovery. What part of drug discovery we can enhance, or we can accelerate, or we can enable using artificial intelligence. So the first thing, so this is just it's not exhaustive list. It is just some key points where we have seen the applications of AI in accelerating drug discovery. Although AI is everywhere now, AI is in the air now, so we cannot escape from it. And each and every step of drug discovery, AI is being used actually. So what we will see is, we will see that AI can be used in drug target identification and validation by predicting the protein ligand interactions, or by prioritizing the target using the AI algorithms, or by doing high throughput screening and virtual compound libraries, performing virtual screening technologies, and then using generative models for compound design, and then doing the predictive toxicology and safety assessment by in silico admit predictions, toxicity modeling with AI, biomarker discovery and patient stratification by using AI in genomics and proteomics, and personalized medicine advancements. So we'll have one by one, we will look at them. So this is a very nice example of identifying a drug target by using artificial intelligence. This is done by In Silico Medicine, a company which is focused on developing medicines using artificial intelligence. So what they have done is, they have taken this omics data, multi-omics data, genomics, transcriptomics, proteomics, epigenomics, metabolomics, signaling pathways, molecular interactions, protein-protein interactions, and then they utilize the text data as well, in the form of like grants or patents or publications or clinical trials or financial reports. And then they put all this data into their AI model, generative AI model. And then with that model, they could come up with several targets. And then those targets were further, screened using different parameters like disease causality, development status, drugability, toxicity, novelty. And then they identified, targets. So they identified 20 different targets for this disease called idiopathic pulmonary fibrosis. And then for these 20 different targets, what they did was they generated novel compounds using generative AI, which we will see later. And then they did primary assays, specificity and selectivity assays, fibrosis assay, collagen production assay. And finally, they have developed this, identified this target, as well as they have identified the ligands or small molecules for this disease, actually idiopathic pulmonary fibrosis. And this drug is in phase 2 clinical trial as of now, and it took only 18 months starting from scratch to getting into the clinical trial for this drug. So second thing we can do is, after target identification, we can use AI in hit identification as well. So usually when we use structure-based approach, for example, if you use docking, so you can dock maybe 100,000, 10,000, 1 million compounds. But if I want to screen like 1 billion compounds through docking, it will take a lot of time and lot of computational resources I need. Okay. But by using deep learning or by using artificial intelligence, what we can do is, like in this case, what they did was they took this, large library, and they took a very small sample, like 1 million compounds. And then they converted them into fingerprints, and then they docked these molecules, and then they made a predictive model using this docking score and the fingerprints. And they used this predictive model to predict the docking score of the huge 1 billion library. And then they could identify small molecules which can be procured and can identified test can be procured and tested in the in vitro setup. So, and this is how we are also using it in our lab to screen ultra large libraries for computational, for screening, for virtual screening, and for identifying targets, for identifying small molecules hits for different targets. And then we can also design molecules using generative AI. So if I ask like ChatGPT, what are different journals in the field of AI and drug discovery, it will give you a nice list of those journals. However, if I ask ChatGPT, can you generate some molecules which are permeable to BBB? So it will not be able to generate because it has not been trained on that kind of data. Okay. But there are tools which can be used, which can generate molecules. And such tools are, for example, these are called as de novo molecular design tools, which can automatically propose novel chemical structures meeting desired molecular profiles, elicit a desirable biological response while maintaining acceptable pharmacokinetic properties. These are also known as generative chemistry, which reflects the rise of generative models in AI. And these models are trained on large data sets and learn to produce output that is similar to the examples they were trained on. So if you look at the molecular design approach, so there are three approaches. One is basically two approaches. One is called direct approach and one is called as inverse approach. So here you have, for example, functional space of all the molecules, where you have the desired properties like redox potential, solubility, or toxicity. And then you have a chemical space, like all the possible molecules, drug-like molecules. Now you wanted to see the direct approach is you synthesize one molecule and then you test this molecule into this functional space and see what if, for example, if we are interested in solubility. So you synthesize one molecule and then you test the solubility and you will feel that, okay, this molecule is lying somewhere. Okay. But our objective is to get a molecule which is lying here in this center of this space, which is having the highest property. Okay. So the other way is the inverse approach, where we have, the solubility data of maybe 1000 molecules. And then from those molecules, we screen them and we identified some molecules which we can, use for our purpose. That is called as inverse approach. And then another inverse approach is this generative AI approach, where you use the optimization or evolutionary strategies or the generative models like variational autoencoders, GANs, RLs, which can start where you can start with a simple molecule which is not lying in your desired chemical space, but you keep on changing it in order to reach to the desired property space, functional space. Okay, so this is how, or this is where the generative AI tools are being used in inverse molecular design technique. And then Reinvent 4 is one of the example developed by a company AstraZeneca. So this Reinvent 4 is, it has different generator models, molecular generators. It can generate different molecules. It has a scoring function. It can generate the molecules by atom. It can generate the molecules by using libinvent, where it can generate the library design and do the scaffold decoration. Or it can, generate the linkers as well, fragment linking and scaffold doping. Or it can do the mole-to-mole transformer-based molecular optimization as well. And then these molecules, the generated molecules, can be scored on different parameters, like you can use RDKit property prediction, ChemPro property prediction, pharmacophore matching, or molecular docking, or QSAR-based property prediction. And then you can use the reinforcement learning as well. And what is reinforcement learning? So here it works like, on the basis of reward and punishment. Like this generator model is generating one molecule. Okay, and then this generated molecule is being tested by, docking, whether this generated molecule having good docking score. If the docking score is good, then this model will be rewarded. Okay, and then it will be told that, okay, generate similar molecules. And if this generated molecule is having a bad docking score or a bad property, so this model will be punished, and then it will ask not to produce similar molecules. Okay, so that is a way to optimize to keep the good molecules and avoid the bad molecules. Okay, yeah, so and then you can use transfer learning as well, where you can, you have trained a model and then you can use a small subset of molecules, and then you can transfer this information to this model, and so that it can use this information to develop, to generate new molecules. So Chemistry 42 is another tool, another generative AI tool, where the in silico medicine researchers have used it for designing nanomolar inhibitors for multiple targets like CDK20 or DDR1. Then coming to the next part where AI can be used is the toxicity prediction. So there are several toxicity prediction tools have been developed, like the Safet AI, which is predicting 5 safety endpoints like hepatotoxicity, carcinogenicity, mutagenicity, nephrotoxicity, cardiotoxicity. Okay, and then there are these tools called Deep DLE, Deep Carc, Deep MS. So Deep DLE is where it can predict whether a molecule is going to have liver toxicity or not. DLE is, by the way, drug-induced liver injury. Okay, so and then this is how this Deep DLE model was developed. And then this is how the deep carc model is being developed. And this is another tool called as skin doctor. So where it can predict whether a molecule will be, skin sensitizer or not. So by the way, how much time do I have, Mishra madam? Do I have like?
Yes, sir, you have enough time.
Okay, good. Thank you. I'll take maybe 15 more minutes actually.
Sure, sir. Sure, sir.
So then you have the deep hit, where it is a Hurge-induced cardiotoxicity prediction model, where it can predict whether a molecule is going to be, inhibiting the Hurge ion channel, which is one of the major cause of a failure of drugs in late stage clinical trial. There was a drug called rofecoxib, which was a COX-2 selective inhibitor, and it was withdrawn from the market. It was approved and being used in clinic for as an analgesic for COX-2, inhibiting COX-2, but it was withdrawn from the market because it was having this Hurge toxicity actually. So they have developed these 3 different models, like descriptor-based DNN, fingerprint-based DNN, graph-based GCN model, where where the model can predict whether the molecule are going to be Hurge blocker or not. And then there is predictive ADMET, where you can predict the ADMET properties of molecules. And I have been fortunate to work with Professor Kenji Mizuguchi at Osaka University, Japan, Nihon, Japan, where I worked with him on developing these some of these models actually. So one of the model which they have developed is called as they have developed a tool called as DRIEMS, and then this is one of the model which they have put into DRIEMS, where you can predict the fraction of unbound drug in the brain by using the prediction model. So what they did was they took the data set from Campbell. Campbell is a very good source of data set, and then they have used the KEGG drug data set, and then they calculated several descriptors and mood rate descriptors and pedal descriptors, and made several ML models using random forest, gradient boost, SVM, etc. And then they have validated their models by using statistical parameters such as R-square and RMSE. And this is the platform landing homepage, DRIEMS version 101, 1.1, and then this is a drug metabolism pharmacokinetic analysis platform, where you can just enter your structure of the compound, and then it will tell you whether the molecule will be, how much will be the fraction of unbound drug in the brain. And not only that, they have at least, I think, 15 to 20 different parameters you can predict by using this server. And those all are mostly pharmacokinetic parameters. And then coming to how AI can be used in clinical trials, because clinical trials is another important step in a drug discovery. Because if a drug is failing in clinical trial, it means that the company is going to lose a lot of money, right? Because if a drug is failing in earlier phase, then the investment is not that much. But if it is failing in clinical trial, means the company will lose a lot of money, because it has invested so much of money, so much of time, so much of manpower hours in that. So if we can make some tools which can predict, which can help us to improve the outcome of clinical trials, so that will be really, really useful. And that's a really hot topic nowadays for research and for startups as well. So PyTrial is one of the package. It's an open source package, Python package that implements various clinical trial tasks supported by AI algorithms. So what it does is it can predict the patient outcome. It can do the trial site selection. It can predict the trial outcome. It can do the patient trial matching, trial similarity. It can generate the synthetic data, and this is how it looks like actually. There is a pi-trial architecture, and they're using different models: GNN, graph neural nets, transformer, RNN, and then they have different modules, as I said, like patient simulation, trial outcome, trial data, site selection, trial search, outcome prediction, trial matching, and you can use it for all the 4 phases of the clinical trial. Okay, so this is a really good tool which one can explore for designing clinical trial, for making, for improving the chances of success of the clinical trials. Okay, but everything which is shining is not not gold actually. So there are several limitations. And what are those limitations? So if you look at this news, that Sumitomo schizophrenia drug discovered with AI tech failed in 2 phase 3 clinical trial. So you can just imagine, like, if a company is having a failure in phase 3 clinical trial, then how much money they would have lost, actually billions of US dollars they would have lost, and it has gone into the drains without any outcome. Okay, so nothing is foolproof. So you always have, and especially drug discovery is highly challenging. There are multiple points, multiple things where it can go wrong. So we need to be extra careful at each of the step. And this is the Gartner hype cycle, where once a new technology comes, and this is about the AI. Once a new technology comes, you see a lot of visibility and a lot of expectations from the technology. But as the time progresses, the disillusionment happens, and then the visibility goes down, and then the slope of enlightenment happen, and the plateau productivity comes. So right now we are, I think, somewhere here at the peak of the inflated expectations, and hope we will see the plateau of productivity very soon, where we can see the real world example or real world application of AI enabled drug discovery. Sorry. So, so looking at the limitation, so the biggest limitation is the data quality and availability. So the data is limited in quantity. Okay, especially in drug discovery, obtaining the comprehensive and high quality data can be really challenging, as the experimental data is often limited or expensive to generate. Data heterogeneity is another issue, where the diverse type of data, including genomic, chemical, and clinical data, needs to be integrated. However, these data sets often come in different formats and varying levels of quality. And then the complexity of biological system is a big challenge, because the biological system is really complicated. And then if you if you don't understand the biology carefully, then it can lead to misinformation. And multifactorial nature is another problem, where almost every disease is not only kind of a single target which is working in the disease, but it's a lot of things which are at play, and then multiple pathways, multiple pathologies, all those things are there. And then the interpretability of models. The models are mostly black box in nature. So most of the AI models, especially those deep learning models, they are considered as black boxes, because their decision-making process is not easily interpretable. And then interpretability is very, very important for understanding the rationale behind prediction, gaining insights into the potential drug targets. And then transferability of models, the domain transfer. The models trained on data from one domain may not generalize well to the another, and transferring knowledge between different therapeutic areas or diseases can be challenging. And then data drift is also a problem. Changes in experimental protocol or data sources over time can lead to data drift, affecting the performance of AI models. And computational resource is another big issue, because with the advent of all these AI models and deep learning models, you need a lot of computational infrastructure, GPUs, CPUs. Heavy computational need is there, and then we are lacking somewhere in that as well. And ethical and regulatory concern is also another problem, where the ethical use of AI in drug discovery, including issues related to data privacy, consent, and potential biases. And then regulatory approval, developing AI-driven technology drug discovery method that adhere to regulatory standards and obtaining approval from regulatory agencies was challenged due to the novel and dynamic nature of these technologies. collaboration and integration, because this should be interdisciplinary collaboration where people from different expertise come together and, bridge the gap between these discipline, and integration of experimental work. And of course, the cost and investment, where the resource investment is, developing and maintaining AI-driven drug discovery pipeline requires a lot of investment, and small research institutions and startups may face challenges in securing the necessary resources. With those words, I would like to, say thanks to Mishra Madam as well as Professor Siddhartha for the the invitation and having me on this platform, for sharing what we have been doing. I didn't share what we are doing because I think that I shall introduce how it can be used. And then in addition to that, maybe I can simply say that for those who have not seen it, I can just show that I have created this course on NPTEL, which was offered this summer, and now it is completed, but it will be offered again in next summer in July 2020, 2016, 2026, where it was quite successful actually, with almost 10,000 enrollments and 3,000 people they wrote exam for this course. And I have covered all these important aspects of, how AI can be used in drug discovery and development, starting with what is the basic drug discovery, what is AI, basics of AI, what is how AI can be used in target identification, prediction, validation, how it can be used in high throughput screening and lead identification, how it can be used in lead optimization, in predictive modeling, in clinical trial, in de novo design, and then case studies and some hands-on sessions. I have covered several, I think 7 or 8 hands-on sessions as well, where I've shown by using Google Colab, I have shown how we can use these, these tools to utilize these AI ML tools. So with those words, I'm again thankful to all of you for your time and for your kind gesture. Thank you. If you have any questions, I'm happy to take those questions.
Thank you, Dr. Rajneesh, for giving this insightful session, specifically AI as well as machine learning and deep learning. You have enlightened all our delegates, those who have joined us in all the fields with pros and cons of this system. Here, there are few questions from some students. I would like to ask you if you are in a mode to answer, please.
Yeah, I can read those questions. So Sahishta has asked like from where we can learn AI/ML skills for drug discovery. So I think I have already answered this question. So
Okay.
you can attend such courses. One of them I have created, and then try to attend hands-on workshops. Try to follow people who are active in this area, who are contributing to this area. And the best thing is that most of the, not most of the, but all of the tools, around 90% of the tools which you can use are open source actually. So you don't have to pay for those tools. You just need to learn a little bit of techniques. For example, it is as simple as you go to a restaurant and order a meal and eat it, or you go and cook your own meal. So if you are a good cook, you can cook much better meal than you get it in a restaurant, because restaurant you will get a fixed meal, you cannot have alterations in that. But if you are a good cook, you can make even better meal. So, and for good cooking, you just have to learn the skills. So I would suggest that try to learn the skills, not just blindly follow that, okay, press this button, this button, and then that is done. That approach is not going to work. So you should ask questions. You should understand, you should all the time, you should ask questions like, what is happening, why I'm pressing this button, what is happening in the background, how it is calculating. So if you ask 5 whys, why, so then probably you will get answer to the problem, and then you will be good at doing it. So my suggestion is that attend. And the best thing is, after internet and after this era, everything is in your hand. You have a smartphone, you have everything on YouTube or internet. You have very nice workshops are being conducted internationally at international level, And most of the material of those workshop is they deposit it in either in GitHub or Zenodo or on their own websites. So you can get it from there. They have talks in the YouTube, recorded talks. You can listen to those talks. And then, but first you have to do is to decide what you want to do, and then you have to make a plan and follow that plan, and you have to be consistent and you have to be honest actually. And you have to spend time, of course. That is the way you can learn actually. Okay, and then second step is what can you step by step career path. And I think I have asked both of your questions. And then another question is, we used to hear black box models, data bias due to biological complexity. Please give your input on this. Yeah. So black box model is like, for example, the idea of having deep learning was, or AI was, to understand how our brain works. Okay, because till now we don't know how our brain works, how we are able to solve the complex mathematical things or other challenging questions. But the problem is that now we even don't know how the AI models are working, the deep learning models are working. So the deep learning models, they can give us an outcome, but we exactly cannot say how it is working. So that is why there is development, there is new technique is being developed called as explainable AI, where we try to add an explainability into the AI models. Whenever it is giving some outcome, it will tell you, okay, this feature is important, that feature is important, this is how it is done, how that is done. So I think that is very important to understand. One example I just wanted to give you, like there is during summer, if it is hot summer, then the increase in ice cream sale will be there, right? So there will be increased ice cream sale, and then will be increased, sunburn. Cases of sunburn will be increased. Okay, so if you look at the correlation, so both, there is a very nice correlation between sunburn and sale of ice cream. Okay, sunburn and sale of ice cream. But is there any causation? Is increased sale of ice cream causing sunburn? No, right? There is no causation. There is no relation between these two variables, but these two variables are going up at the same time. So there is a very nice, correlation between them. So we have to look at the causation, not the correlation. We have to understand, and this is where the explainability comes from actually. We can clearly see that increase in hot summer means increased sunburn, because there is heat, and heat is leading to skin, burning, and then sunburn, all those things. So we need to understand this causation, then only we can add explainability to models, and that we can only understand once we know the basics and we know the in detail all those things. So I think yeah, if there is no other question, or if there is any other question, I'm happy to take. Yeah, I think I've answered the questions which were there. Yeah,
Absolutely.
Thank you so much, sir, for your rich presentations on generative AI in drug discovery opportunities and challenges. Thank you so much, sir. Now I request our session chair, Professor Dr. Anuradha Mishra, Madam, to felicitate our distinguished speaker, Dr. Rajneesh Kumar.
Yeah, thank you, sir, for this wonderful presentations and sharing your views, specifically how we can utilize this AI, not only with the artificial tools, but using our own brain, how you can implement, how you can calculate. And I would like to appreciate here you have initiated with the lead molecule, how we can identify the lead molecule, that too what could be the toxic profile. We can not only analyze the toxic profile and all that. So this is a gesture of appreciation. Kindly accept this from School of Pharmacy, DRIEMS University, from the all team members, from the core team members, from the conveners, coordinators. Thank you so much.
Yeah, thank you so much. I am really grateful to all of you for providing me this platform, and I'm sure to see all of you sometime in person as well. I would love to come to Odisha actually. Because,
Sure, sir.
My first PhD student is from Odisha, and he has promised me to invite me on his wedding, so I'll be there sometime in next year.
Definitely, we'll meet, sir. We'll meet.
Yeah. Okay. Thank you so much then.
Thank you so much, sir.
May I take the leave now?
Yes, sir.
Yeah, you may leave, sir.
Okay, thank you. Bye-bye.
Thank you,
See you.
Thank you, sir. Thank you so much, madam, for your gracious presence and support on this scientific session. Now, I request our convener and Dean School of Pharmacy, DRIEMS University, Professor Dr. Amulya Ratna Behera, sir, to felicitate our session chair, Professor Dr. Anuradha Mishra, madam.
Thank you, madam.
Thank you so much, sir.
Thank you for
It's
your
really
valuable
a
time.
pleasure to be a part of this nice and wonderful conference. Thank you so much for inviting me. Thank you, Dr. Siddhant, and thank you all the team members, conveners, for giving me this opportunity.
All pleasure mine. Thank you, madam.
Thank you so much, sir. As we conclude the second day of this enlightening conference, I would like to express my heartfelt gratitude to all the speakers and the session chairs for sharing their immense knowledge and expertise with us. I appreciate all the attendees for their active participations and valuable contributions. Please do not forget to submit the feedback form. It's been a pleasure hosting this conference, and I hope everyone leaves us today with a great understanding and new perspectives on our focus topic. Without any further delay, we will switch to the closing ceremony and presentations of our to the e-poster winners. Over to the moderator of the closing ceremony, Sarada Kanta Sahu, sir, Associate Professor, School of Pharmacy, DREAMS University. Over to you, sir. Thank you.
Let us please check whether all delegates have joined in the platform.
Yes, sir. Yes, sir. We are checking.
Siddharth, sir.
Yes, sir. Please.
Should I call to Vice Chancellor, sir?
Yeah, Vice. We are waiting to Vice Chancellor, sir.
Okay.
Listen, sir. Am I audible? Alok sir have joined. Also convener. Join कर सालेनी.
Sir, BC sir also joined.
BC sir is just on the way. He's coming.
Madam, start. My BC sir is there. Madam, please.
A very good afternoon to one and all. Namaskar. I, Saradakanta Sahu, Associate Professor, School of Pharmacy, DRIEMS University, welcome you all to this prestigious gathering. As you know, we stand at the fascinating intersection of artificial intelligence and pharmaceutical sciences, a junction promising to revolution how we discover, design, and develop life-saving medicines. It's an honor to host you on this validatory session for the DBT-CTEP-sponsored National Online Conference on AI-driven design and development of new chemical biological entities and formulation approaches, challenges, and road ahead. This three-day intellectual feast, organized by the School of Pharmacy, DRIEMS University, brings together leading scientists, researchers, academicians, and students from across the nation to deliberate on the most transformative area of drug development today. So let us commence this remarkable journey with a traditional and auspicious start, the invocation song. May I request our faculty, Ms. Rosalind Maharana, for this invocation song.
नीलाद्रो शङ्खमध्ये शतदलकमले रत्नसिंहासनस्थं सर्वालङ्कारयुक्तं नवघनरुचिरं संयुक्तं चाग्रजेन भद्राय वामभागे रथचरणयुक्तं ब्रह्मरुद्रेन्द्रवन्द्यं वेदनां सारमीशं सुजनपरिवृतं ब्रह्मदारु स्मरामि ब्रह्मदारु स्मरामि ब्रह्मदारु स्मरामि. Thank you.
Thank you so much, Rosalind ma'am, for this beautiful song. May this devotional song illuminate our minds with knowledge and innovation. We are truly honored by the presence of our distinguished dignitaries. I would like to invite our respected Dean, Dr. Amuliratna Behera sir, Dean of the School of Pharmacy, to deliver the welcome address and set the context for the conference. Sir, now the screen is yours, sir.
Namaskar, Honorable Vice Chancellor, esteemed Dean Research, Dean IQAC, Director IQAC, Convener IIC Cell, Coordinator of this conference, participants, fellow faculty members. Sir, I would like to congratulate Professor Siddharth Shankar Kara to receive this grant from Department of Biotechnology, CTEP, Government of India, and to organize by putting lots of effort, and the organizing team is very much rigorously observing these online conferences. Hope these conferences added many more knowledges or insights for their future research. Thank you. Thank you all.
Thank you so much, sir, for your valuable word. Now, I would like to request our IIC convener, Dr. Deepak Ranjan Biswal, sir, for the address the gathering. Sir, please.
Thank you. Am I audible?
Yes, sir, you are audible.
A very warm good afternoon to you all. Honorable Vice Chancellor, DRIEMS University, Professor Dr. Prakash Kumar Hutta sir, Director Coordination, Mrs. Chinmay Ratha Madam, Director IQAC, Professor Dr. Alok Ranjan Biswal sir, Dean Research, Professor Vishwaranjan Mahanti sir, Dean School of Pharmacy, Dr. Amulya Ratna Behra, sir, Dr. Siddharth Shankar Karo, coordinator of this particular conference, distinguished speaker, guest, faculty members, and student participants. As the convener of the IIC Institution Innovation Council DRIEMS University, it is my privilege to deliver the closing remark of this 3-day DBT-CTEP Government of India sponsored National Conference on AI-driven design and development of new chemical or biological entities and formulation, and the roadmap. Over the past 3 days, we have witnessed a rich exchange of knowledge and ideas, bringing together eminent academicians, industry experts, and researchers to explore the transformative impact of artificial intelligence on chemical sciences, biological discovery, and pharmaceutical formulations. At IIC, that is the Institution Innovation Council DRIEMS University, we are committed to building an innovative driven ecosystem that embraces artificial intelligence as a catalyst for progress. Through AI-focused workshops, hackathon, and expert interaction conferences, we empower students, researchers, academicians to think beyond conventional boundaries and strengthen academia-industry collaborations. This conference has further highlighted how AI-driven interdisciplinary innovation is shaping the future of chemical, biological, drug development, and pharmaceutical sciences. My heartfelt appreciation also in this particular event goes to the organizing committee, all the technical committee, faculty coordinators, student volunteers for their dedicated effort in ensuring the smooth conduct of these online conferences. As we conclude, let us take forward the ideas, collaborations, and inspiration generated over these 3 vital days. The road ahead for artificial intelligence in chemical and biological sciences holds immense potential, and together we can contribute meaningful to this evolving landscape. Thank you all for making this conference a grand success. Thank you. Thank you all.
Thank you so much, sir. Your words resonate deeply, sir. Thank you so much.
Thank you.
Now, to discuss the critical aspects of quality assurance and continuous improvement in this AI-driven era, I would like to invite our Director IQAC, Dr. Alok Ranjan Biswal, sir, to deliver his address. Sir, please, sir.
Thank you, Santa sir. Am I audible?
Yes, sir.
Okay, thank you very much. Honorable Vice Chancellor of DRIEMS University, the convener and co-convener of this particular national conference, the esteemed guests, distinguished speakers, delegates, faculty members, and dear participants. So as we come to the close of this enriching 3 days national conference, it gives me an immense satisfaction as well as pleasure to reflect on the depth of the discussions and the quality of knowledge shared. Over the last 3 days, we have witnessed a remarkable convergence of ideas ranging from artificial intelligence-enabled drug discovery and computational modeling to breakthrough approaches in formulation science and bioengineering. From the IQAC's perspective, this conference beautifully reflects our institutional commitment to fostering a culture of quality, collaboration, and forward-looking research. The interdisciplinary spirit displayed here reinforces the importance of integrating emerging technologies with core scientific disciplines to address the global health and the industrial challenges. I extend my heartfelt appreciation to all the keynote speakers, the panelists, the presenters, and the participants for their invaluable contributions. A special word of gratitude goes to the organizing committee members whose hard work and meticulous planning ensured that every session was meaningful, engaging, and impactful. As we conclude, I hope the dialogues initiated here will continue to grow into the fruitful research partnerships, innovative projects, and academic excellence. Let us carry forward the momentum generated in these 3 days and continue to advance science with responsibility, creativity, and purpose. Thank you all, and wish you all the success in your future endeavors. Thank you all.
Thank you so much, sir, for sharing your perspectives on the future of research. Thank you so much. Following that inspiration address, now I would like to invite the driving force behind our research initiatives, the Dean Research, Dr. Vishwaranjan Mohanty sir. Sir will shed the light on how AI is strategically integrated into our institutional research roadmap. Sir, please join with us.
Thank you, Dr. Sarada. Honorable Vice Chancellor, Professor Dr. Prakash Kumar Hota sir, Director Administration, Srijukta Balaram Karo sir, Director Coordination, Chinmay Madam, Director IQAC, Professor Dr. Alok Ranjan Biswal sir, Dean Pharmacy, Professor Amulya Ratna Behera sir, IIC Convener, Dr. Deepak Ranjan Biswal sir, and our coordinator, Professor Siddharth Shankar Karo sir, esteemed dignitaries, distinguished speakers, invited guests, faculty members, researchers, and dear students. A very good evening to all. It is really an honor and a privilege to deliver the closing remarks for the enriching 3 days academic events. What began as an idea to create a platform for dialogue on AI-driven drug design and modern pharmaceutical innovation has today taken the shape of a meaningful and impactful scholarly exchange. Over the past 3 days, we have witnessed dynamic technical sessions, thought-provoking keynote addresses, and insightful interaction that had truly broadened our understanding of how artificial intelligence and machine learning are transforming chemistry, biotechnology, and pharmaceutical sciences. From computational drug design and bioinformatics pipelines to predictive modeling, formulation optimization, and regular consideration, every session has added depth and clarity to our collective knowledge. We have more than 405 participants registered from 16 states of this country, 150 faculty member, 82 research scholar, and 172 students have registered in this conference. We had 6 brainstorming sessions and 4 e-poster presentation sessions. Out of 60 posters presented, 6 posters got selected for the award. The satisfaction level of participants is reflected in their feedback. All the credits, I sincerely acknowledge the efforts of the entire management of DRIEMS University, organizing committee, technical committee, scientific committee, and supporting teams. Their dedication from planning and coordination to logistics and execution has ensured the smooth conduct of these 3 days events. All above, I extend my warmest gratitude to all the participants and delegates who joined us from various institutions across the country. Your enthusiasm, active engagement, meaningful questions, and willingness to share ideas have been the true strength of this conference. As we conclude this national conference, we take with us not just knowledge, but new perspectives, collaboration, and a reinforced commitment to advancing pharmaceutical and biotechnological research through the power of artificial intelligence. Let us continue to innovate responsibly, explore fearlessly, and contribute meaningfully to the scientific community and the nation. On behalf of the School of Pharmacy, DRIEMS University, I once again thank you, each one of you, for making this conference a highly successful and inspiring event. Wishing you all a safe journey back and a bright future ahead. Thank you. Thank you all.
Thank you so much, sir, for that insightful overview. Your words deeply resonate to us. Thank you so much, sir. Wonderful, sir. Now, it's my immense privilege and pleasure to introduce and invite a visionary leader, the guest Vice Chancellor of DRIEMS University, Professor Dr. P.K. Hota sir. Sir's guidance has been instrumental in fostering a culture of research and innovation. We are eagerly look forward to his special address. Sir, please.
Good afternoon to all of you. Is it audible? Is it audible?
Yes, sir.
Yes, sir.
Absolutely
Yes,
audible.
sir.
It is audible.
Absolutely audible, sir.
So after 3 days program, I learned from the Dean research that completely all the objectives have been fulfilled in shape of poster presentation, keynote address, then the technical sessions, paper presentation, also paper presentation. So knowledge, assimilation and dissimulation. These are the two aspects for which this conference has been organized. So this is the IIC initiative which has been organized by the pharmacy department. So pharmacy department is conducting this kind of seminar, workshop, expert lecture, then the industry visits, all those things are happening. It is a regular feature. So organizing this conference is another addition to this. I am happy that the way the IIC is advancing with the support of all the schools, so that our accreditation and our visibility will be reaching. And the people who have attended from various parts of the country, at least they came to know that there is a DRIEMS University, multidisciplinary university, who is arranging all those things. Particularly, I expect the candidates or the participants who have already attended all these course, all these sessions will be definitely benefited. Why I am telling it? Because today the technology is evolving. Pharmaceutical science or the education is not an exception. So I've already discussed with the deans and the faculty members of the pharmacy school. So the restructuring of the pharmacy education is another demand. And slowly, we are incorporating, particularly keeping in view the PCI requirements and the university's objective. Probably you'll be getting, those who will be joining in the first year, they'll be seeing that 2 courses on computer, including this machine learning and artificial intelligence at appropriate semester, particularly 3rd and 5th semester has been incorporated. Machine learning and artificial intelligence starting from the school level, this is a national vision, not the university vision. So it is somehow the mandatory thing. My observation is that though it is technical thing or the technology you can say, still you can find that most of the difficult tasks which you are doing by analytical methods or by graphical method or by, let us say, instrumental methods also, you are not happy that whatever you are thinking, that cannot be done. For example, modeling. For example, optimization. For example, identification of the parameter. For example, the estimation of the parameter. All those things are also needed in pharmacy education also. So conventional method or the experimental method, it is one side. Second side, if you want to realize whatever you are doing research and all, so the help of this machine learning as well as this deep learning or the artificial intelligence, it evident. You cannot get rid of that. So point to be noted here is that particularly in pharmacy education, what I understand, this optimization task has to be done because the drug formulation, let us say drug therapy also people are talking about those things. Then those things can be easily understood by the researcher and it will be conveyed to the what you call the students also, undergraduate also. Because NEP 2020 talks about that research must be integrated at the undergraduate level Earlier, what was happening? Earlier, people are thinking that you do the masters, then there's something called MPhil and all. Then you think of research. No, research is integrated into the undergraduate level. How will do that? So those particularly faculty members, those who are teaching to the undergraduate students or the postgraduate students, so they should in every class, in addition to the content they are teaching to the student, you just spare 5 minutes regarding the research. Whatever topic you are doing, how the research can be done in this particular topic, if you will tell that in terms of a story, then also student can pick, because today's pedagogy means the way of teaching to the student can be storytelling based, can be art integrated, can be sports integrated. So mostly student will be happy if a teacher can think or he can incorporate his research idea through a appropriate story, then probably student will accept that, and definitely some interest will be inhibited into that. So teacher's role, I expect that should be done. And at the end, I expect one or two participants should interact with us, or they can give the feedback. Of course, online they will be giving the feedback. Now I request the coordinator, Professor Kar, is it possible that two of the participants who are just now online, can they speak something?
Sure, why not, sir? We may ask our moderator. Can you please invite any two participants to speak voluntarily?
Okay. After then, we will conclude.
Sure, sir.
You just bring two participants to the.
Yes, sir. We'll just try.
Yeah.
Dear participants, if anyone is interested to speak any your comments on this particular program or your feedback, can you please speak up by turn on your mic?
Yes. Can
please.
I speak?
Yeah, sure, sir.
Okay. Very good evening. Myself, Dr. Ashwini Kumar Senapati from IPT Salipur, Institute of Pharmacy and Technology Salipur. Actually, the theme of the conference is very nice because this is the need of the hour. Actually, we are not that much aware of the AI tools, how it is implemented in the drug discovery process. It should be in offline and hand-on practice. That means one by one, or it may be done with a additional workshop, so that it will be much more fruitful and beneficial to us. Thank you. And every talk was very nice and very much integrated with the new drug development process, which is needed for the hour. Thank you very much. Thank you very much for giving scope to share a few words.
Okay, Professor Kar, you just note it down. In our future,
Yes, sir, it's already recorded. Yeah,
we'll
it's
be
recorded,
definitely
sir.
adding to that. Okay.
Yes.
Thank you so much, Professor.
Thank you so
Anyone,
much, sir.
Anyone, please? Any other participant? If you have any input, can you kindly turn on your mic and speak? Any research scholar, any PhD scholar, please? We have around 85 PhD scholars registered.
Yes, sir.
Yeah,
Can
please.
I, sir?
Yeah,
Yes,
you're
sir.
audible.
Yes, sir. I am Shri. Yes, sir. I am Mandali Srividya. I'm from JSS College of Pharmacy, Ooty, sir. I'm a PhD scholar, and these sessions were so informative, especially the discussions on real-world applications of AI and future impact of emerging these technologies. It's so useful to us because I'm entered into PhD. I was in first year now. It's so helpful, and the speaker presented complex topics in a clear and engaging way, which made the content accessible even for attendees who are not deeply in technical way. So it is so helpful to us who are coming into the PhD for research. It is so helpful, sir. Thank you so much.
Thank you, madam.
Thank you.
So,
Thank you so much.
professor and graduate, our research college over. Any postgraduate student? If you have any feedback, please. Any postgraduate students? We have around 180 students registered. Okay. Okay. It's over to moderator. Please unmute yourself, Sarda sir.
Thank you. Thank you so much, sir. Thank you, DC sir, for sharing your perspective on future of research. Thank you, sir. Now, I am going to declare the result of e-poster present competition. Sir, am I audible? See that, sir?
Yes, sir. Absolutely audible.
Out of 60 e-poster presented in 4 online sessions on yesterday, the external evaluator selected 6 e-poster. All the participants are doing their best and elaborate so nicely. It is very difficult for our evaluator that how can they select and judge them. But as it is a competition, we have to choose the best one. It is only 0.5, 0.2 differentiation, but it is very difficult task for our external evaluator. Let me declare the name of the winner. Code number SOP PC20 Mali Sanket Sambhaji, Yashoda Technical Campus, Faculty of Pharmacy, Satara, Maharashtra.
Okay. Congratulations to Mr. Mali.
Second one is SOP code number PC25 Chetan Milan Rajkot, Department of Pharmaceutics, YV Chavan College of Pharmacy, Maharashtra.
Okay. Congratulations, Chetan.
Next.
Thank you, sir. Thank you so much.
Thank you. Code number CG07 Meenakshi Manekar, Nutan Ayurvedic College and Research Center, Mehsana, Gujarat.
Many congratulations, Meenakshi.
Next, code number CH08 Sagarkant Mohanty, JSS College of Pharmacy, Uti.
Okay, congratulations, Sagarkant Mohanty.
Next, code number PC01 Debasis Misra, School of Pharmacy and Life Science, Centurion University of Technology, Bhubaneswar, Odisha.
Thank you, sir.
Okay, congratulations.
Thank you, sir.
Yes, yes.
Next, code number SOP CL09 Athira BK, Nite DU, NJSM Institute of Pharmaceutical Science, Mangalore, Karnataka.
Nite University.
Okay, congratulations, Athira.
Thank you, sir. Thank you.
Thank you, everyone. Everyone gets with their certificate and memento through post. Now, I would like to request our Professor Dr. K.C. Panda for the vote of thanks.
Yes.
Good afternoon, respected Vice Chancellor, esteemed dignitaries, distinguished speakers, honored special chairs, dear colleagues and participants. It's my privilege to deliver valedictory vote of thanks on behalf of School of Pharmacy, DRIEMS University, as we conclude this DBT-CTEP sponsored online conference. At the very beginning, I extend our heartfelt gratitude to the Department of Biotechnology DBT for sponsoring and supporting this academic initiative. I express my deepest appreciation to our eminent speakers who enriched today's conference with their expertise and thoughtful presentations. Dr. Sairam Kalapatapu, Dr. Samir Agrawal, Dr. Rajnish Kumar. Thank you, esteemed speakers, for your scholarship scholarly contribution and for sharing cutting-edge knowledge with our participants. We are equally grateful to our distinguished session chairs for guiding the academic session with excellency, Professor Dr. A. Ramakrishna, Professor Dr. Amiya Kumar Pushti, Professor Dr. Anuradha Mishra. Your leadership, insightful comments, academic wisdoms added great value to the conference. We are especially grateful to our Honorable Vice Chancellor Sir, Dr. P.K. Hota Sir, for his continuous guidance, motivation, and unwavering support. My sincere gratitude extend to the respected leadership of DRIEMS University, Dr. Pramod Chandra Ratha Sir, Chairman DRIEMS University, Sri Balaram Kara Sir, Director Administration, Dr. Chinmay Ratha Madam, Director Coordination, Dr. Deepak Biswal, Convener IIC, Dr. Alok Ranjan Biswal, Director IQS. A special word of appreciation to Dr. Amulyaratna Behera, Dean cum Professor, Department of Pharmaceutics and Convener of this conference. Your tireless efforts, exceptional coordination ensured the smooth execution of the program from start to the end. Dr. Siddharth Sankar Kara, Professor, Head, Department of Pharmaceutical Chemistry and co-convener of this conference. Dr. Kara deserves special mention for his pivotal role in arranging and bringing the DBT-sponsored program to the School of Pharmacy, DRIEMS University first time. His proactive involvement from planning the technical session to coordinating the speaker, managing communications, ensuring academic rigor has significantly elevated the quality of this conference. His leadership, enthusiasm, and tireless effort behind the scene contributed tremendously to the seamless execution of the program from the beginning to the end. Special acknowledgement to the Dr. Vishwaranjan Mohanty Sir, Dean Research, DRIEMS University, for its continuous guidance, research leadership, and valuable support in facilitating smooth organization and successful conduct of this program. And special acknowledgement entire organizing team, especially Mr. Harish Mishra Sir, Ms. Kritika Mohanty, Ms. Gargi Mishra, Ms. Rosaline Maharana, Mr. Sardakanta Sahu, Mr. Somiranjan Bhadai, Mr. Dr. C.S. Barik, HOD Pharmacology, Suryakanta Panda, Vincent Sahu, Vinod Behera. Your commitment, meticulous coordination, technical support ensure the seamless execution of these online sessions. To all researchers, faculty members, students, attendees, thank you for your enthusiastic participation. Your engagement made this conference truly meaningful and successful. As we conclude, let us carry forward the knowledge, inspiration, and collaborative spirit gained today. May this conference encourage further research, innovation, and academic growth in the field of pharmacy and biotechnology. Thank you all once again. Wish you success, good health, bright professional journey ahead. Jai Hind, Jai Bharat. Thank you.
Thank you all. Thank you.